Hi everyone, good evening guys. Welcome back to Unacademy NEET English channel. My name is Tina and we are back once again in Re-NEET series guys, and today we are going to start with class 12 NCERT reading. But before we start with that big thing, there's another big news for you guys. For your Re-NEET preparation, we are bringing to you this amazing offer just for rupee 1. You're getting 25 tests in total. 10 minor, 6 subject-wise tests, 3 major and 6 overall tests. Please don't let even a single day go without you taking these tests, these mock tests, and you can take them at your convenient time. Although, as always, my strong suggestion is to set your biological clock for 2 to 5 p.m. only. So try that you take these tests every day. I hope you all have signed in, you all have enrolled for this test series, and if you're not done that, not a good thing, not a good sign. Please do that, guys. And also, apart from this Unacademy All India NEET test series, we have absolutely free these mock tests going on thrice a week. So don't forget to take these tests also every Wednesday, Friday, and Sunday, right? But most importantly, take these tests because these tests are going to cover the entire syllabus, detailed result analysis, so you can actually get to know where you are standing. And then side by side, please keep attending these lectures, watch the marathon sessions wherever necessary, redo that and revise your NCERT with me. So on that note, let's start with the class 12 NCERT today, guys. We are going to start class 12 NCERT, starting with the first chapter. Please give me a thumbs up that you're all ready and you have also gone through class 11. You have not missed any session from class 11 NCERT, and we can, yes, and you are very confident about it. So this is class 12, first chapter, sexual reproduction in angiosperms. Give me a thumbs up. You're all set with your NCERTs. Yes, please. Quick. Everybody, please let me know. Let's start this session with a lot of energy, guys. All right, Roshan, I'll do that today. There was some issue with my phone, guys. My phone was not with me. I'll do that today. Okay? Yeah. I'll do that today. Don't worry. All right, guys. Let's start with the first chapter, that is sexual reproduction in angiosperms. And from here, just a second. Let me take my pen. Yeah. All right. Starting here. So this is the first. Wait. Okay, yes. So here we are. All right. So let's start from stamen, microsporangium, and pollen grain, because about flower we have already read in the chapter of morphology. So the first, see, this was 2016 question. The proximal end, we know that stamen is known as microsporophyll, right? It's a modified leaf into a floral structure which gives rise to male spores. So microsporophyll, right? That is another term for stamen, right? Then, so stamen has two parts. One is filament, the stalk is filament, and on the anterior part is present the anther. Now see, so terminal part has bilobed structure called anther, the proximal end of the filament. So this is the proximal end, this is the distal end of filament. The proximal end is attached to thalamus. This was a line in 2016. This was a line given in 2016. Please keep on marking these lines, okay, and or the petal of the flower. Next is in each of this. So a typical anther is bilobed with each lobe having two theca, hence dithecous, right? Now an anther is four-sided structure, so tetragonal, with 4 microsporangium located at corners, 2 in each lobe. So tetrasporangiate, right? And microsporangia later develop into to become pollen sacs. When it will be filled with pollen grains, it will be known as pollen sacs. Coming on to the structure of microsporangium here. See, 2016 question. So a typical microsporangium appears circular in outline with 4 layers. And what are those 4 layers? Epidermis, endothelium, middle layers and tapetum. The outer 3 layers help function is protection and help in dehiscence of anther. That was 2016. The innermost layer of tapetum, it nourishes. The function of tapetum is nourishment. Cells of the tapetum possess dense cytoplasm and generally have more than one nucleus. 2025 question. Now it came in the form of assertion reason. Why does it have more than one nucleus? Because it increases the nourishing efficiency. This is the correct reason. So this was the assertion reason in 2025 last year. Can you think of how tapetal cells could become binucleate? Just by continuing the karyokinesis, but no cytokinesis. That could be the only reason. That could be the only way in which a cell becomes multinucleated. Now the anther is filled with compactly arranged homogeneous cells called the sporogenous tissue in the center of the microsporangium. Let's talk about microsporogenesis. The cells of sporogenous tissue undergo which division? Meiotic division to form microspore tetrads. What would be the ploidy of the cells of tetrad? Please let me know in the chat. Yes guys, what will be the ploidy of the cells in the tetrad? Yes guys. Tetrad, what is the ploidy of the cells? Because it is going to undergo meiosis. So the ploidy will be half of the parent cell, which is N. So the microspores are haploid. They are haploid, while the cells of sporogenous tissue are all diploid. So this is outside to inside. You should know the correct sequence to arrange the layers. Epidermis, endothelium, middle layers, then tapetum, and inner to that are present the microspore mother cell. So each cell of sporogenous tissue is MMC, microspore mother cell. The ploidy of microspore mother cell is 2N, but after meiosis, microspores and microgametes, they will be haploid, right? Now, this process of formation of microspores from pollen mother cell is called microsporogenesis. And as a result of meiosis, it's always 4 cells. So cluster of 4 cells, microspore tetrad is formed. Tetrad is when the 4 cells are held together. Later on, there's a layer called callus which is present outside the tetrad, holding them together. It gets dissolved and the cells separate. Is it clear? See, later on they will separate and develop into pollen grains. Now in a microsporangium, several thousand of pollen grains are formed and released. Why do we need such a large number? Because of the chances of high wastage. Is that clear? Now pollen grain, underline, represents the male gametophyte. Very important. What is a male gametophyte here? Pollen grain. And specifically it is a 2 and 3 cell stage. Now see, if you touch the open anthers of hibiscus, you will find a deposition of yellow powdery substance. We know that. So if we scan it under the electron microscope or even under some light microscope, we may not be able to observe that many details under the light microscope, but electron micrographs will show you a lot of details about the sculpturing present on the surface. See, there are different sculpturings. The sculpturing is very different from in each species, and it is a characteristic feature as well. Anyways, so pollen grain generally spherical, size 25 to 50 micrometer, 2 layered. The outer layer is exine, inner layer is intine. Exine is made up of sporopollen in 2016 question. One of the most resistant organic substance, can withstand high temperature, strong acid, alkali. No enzyme can degrade it. And because of this such resistant nature of sporopollen, that see, pollen grains are well preserved as fossils because of sporopollen. And who secretes it? It is secreted by tapetum. This was asked in 2024. Please make sure that you are revising these things with me very carefully. Yes, please. Is that clear to everyone? Is that clear to everyone? Please let me know. Yes, please. Just a second. Everybody is here? Mm-mm. Yes, please. Let's move on to the next now. So there is a special region where sporopollenin is absent. So pollen grain, exine, has a prominent aperture called germ pore where sporopollenin is absent. So the place in the exine where sporopollenin is absent, that is known as germ pore, and it is needed for the protrusion of intine as a pollen tube. Is that clear to everyone? Clear to everyone? Please let me know. Yes, please. All right. Now, moving on. So, the inner wall of the pollen grain is made up of intine, which is thin and continuous and made up of cellulose and pectin. So, exine is discontinuous because there is a germ pore present where sporopollenin is absent, right? While intine, made up of cellulose and pectin, is continuous. Also, because it is cellulose and pectin making it, so basically intine represents the true cell wall, right? Now, when the pollen grain is mature, it contains two cells. What are the two cells like? The two cells are vegetative cell and the generative cell. So this two cell stage of pollen grain, right? This is the stage at which most of the pollen grains are pollinated. 60% of angiosperms will show pollination at this cell stage. Vegetative cell is bigger and abundant in food reserve and largely irregular shaped nucleus. Generative is small and floats in the cytoplasm of generative cell. Now the point is, why does it lead to formation of unequal sized cells? It is because of asymmetric spindle formation. So that is why one is larger, another is smaller. See, 60% angiosperms will be shed, pollen grains are shed at two cell stage. Remaining will undergo pollination after this generative cell has also further divided by mitosis to form two male gametes, right? So generative cell will divide mitotically to give rise to male gametes. Vegetative cell will not undergo further division. Pollen grains are shed three cell stage before, so that happens only in few cases, in 40% of the sperms, sorry, 40% of the pollens, while 60% of the angiosperms will be pollinated at two cell stage. Now this pollen grain at three cell stage, read this line very carefully, this was 2016 question, one of the best papers of NEET. So represents two generations, one within the other. Okay, so it represents two generations, one within the other. Pollen grains of many species cause several allergies, severe allergies. So some people are allergic to sporopollenin. So exactly speaking, people who are allergic to pollen grains are allergic to this sporopollenin. Is that clear? So it may lead to symptoms like chronic respiratory disorders, asthma, bronchitis, and hay fever you've heard of, right? Is that clear? So the pollen grains which are particularly very allergic in nature, they belong to the category of, to the genus of Parthenium or carrot grass, which came into India as a contaminant along with imported wheat and became ubiquitous. Ubiquitous means found everywhere. Now this is an alien species. Okay, carrot grass, Parthenium. We have already read about alien species invasion. So this is an alien species which is also famous for causing respiratory disorders, that means the pollen allergy. Is that clear to all? Now moving on to next, pollen grains, they are taken as supplements because they are rich in nutrients, and usually it is for increase in the performance of athletes and race horses. Is that clear? Now coming on to the pollen viability. Pollen viability means the time period between the shedding of the pollen grains and their germination. So that time duration. See, so it differs. Period within which pollen grain remains viable is highly variable. So it depends upon the prevailing temperature as well as humidity. Now see, this viability is different. So viability in case of rice and wheat, write them separately. In rice and wheat, it is just 30 minutes. Within 30 minutes, it has to germinate or it will lose the viability. While in some members like Rosaceae, Leguminosae and Solanaceae, Saral, just remember it as SRL, Saral, easy to remember, Saral. So Solanaceae, Rosaceae and Leguminosae, they maintain viability for few months. This was asked in 2023. Is that clear? So match the following statement based questions, direct. So viability for rice and wheat is within 30 minutes, and for these three families, Rosaceae, Leguminosae and Solanaceae, it is within few months. Is that clear? Now if you want to increase the viability, very simple, cryopreserve it. Put it in liquid nitrogen at minus 196 degrees Celsius. So that cryopreservation is going to increase the viability. You store the pollen grains at this temperature, minus 196 degree, like it is done in pollen banks, similar to seed banks. In crop breeding programs, we employ this technique. So why do we do that? To enhance the pollen viability. Please tell me these points are clear. This is done to enhance the pollen viability. Dilip, what should I tell again? Yes, please. What do you want me to say again? Please let me know. All right. Now coming on to the female part, that is pistil. Pistil also known as megasporophyll. Megasporangium is another term for ovule, and then the embryo sac. Now see, these are the examples that you must focus on. If there is single pistil, we call this condition as monocarpellary. We have talked about it in morpho also. More than one pistil will be known as multicarpellary, simple. Bicarpellary, tricarpellary, or simply multicarpellary condition. Now if the pistils are fused, we'll call it syncarpous. If they are all free from each other, that condition is called apocarpous condition. Each pistil has three parts, stigma, style, ovary, we know that, right? We also know that the basal part of the pistil is called the ovary, and the space around the ovule, that is called ovarian cavity or locule. Now placenta we have discussed, and the placentation as well. Now below this figure, guys, there is some important information written here. Please focus here. See, this is A. A is the dissected flower of hibiscus showing the pistil. So it is a stigma which has been split into 5. We call it pentaphyid stigma. It is a character of Malvaceae, characteristic of hibiscus, belonging to the family Malvaceae. Style, and then this ovary lying on the thalamus. Now the point to be noted here is multicarpellary condition. This represents multicarpellary condition and syncarpous pistil of papaver, poppy plant, which gives us opioids. Is that clear? So papaver has syncarpous, multicarpellary, syncarpous condition. Example is papaver, very important. Then multicarpellary but apocarpous condition, that is seen in Michelia. This is Michelia. So multicarpellary but free from each other, that is Michelia. And this is a typical anatropous ovule. Anatropous means it is inverted. Now these examples are important. Papaver has multicarpellary and fused condition, that means syncarpous, while in Michelia it will be free carpus. Please note down these things. Clear? Now arising from the placenta is the megasporangium, also known as the ovule. See another examples here. So number of ovules in an ovary may be one ovule per ovary. Example is wheat, paddy and mango. Paddy is rice, wheat, rice and mango, that they have one ovule, while many in papaya, watermelon or in orchids. So this is the season of watermelons, and we know how many seeds irritate us, right? So there are many seeds in papaya, even in watermelon. Is that clear? So there are certain things that you can easily learn by observation. We have been eating fruits all our life, right? Okay, now just relate it and learn. Talking about megasporangium, another term for ovule. See here, the body of ovule fuses with funicle in the region of hilum. 2020. I've written the PYQ side adjacent to it. Now hilum represents the junction between the ovule and the funicle. See this diagram. So this is hilum. This is the place where this funicle is binding to the ovule, joins with the ovule. Now each ovule has two protective coverings. So that is a typical nature of angiosperm ovule, that it has two integuments. Usually it is two integuments, so bitegumate, right? Now it is filled with nucellus, except at the tip where there is a small opening known as micropyle. Now opposite to the micropyle is chalaza, and chalaza represents the basal end. Now very important. See, it was supposed to be like this. This is a straight ovule. This is called orthotropous. So on the upper side we had this micropyle. So micropyle is always known as the upper end, and on the lower side we have chalaza. So this represents funicle, and the funicle which binds to the body of the ovule, this region is hilum. Do you understand that? Right? But now instead of being straight, in around 82% of angiosperms, this ovule has become inverted like this. Otherwise it was supposed to be this straight, now it has got inverted. So the micropyle has come near the funicle, but we still use the same terminology that micropyle is the upper end, and see, chalaza is always considered as the basal end. Please take notice of this. Is that clear? Please remember that. So we are going to use the same terminology here as well. Yes, please. Clear to all? All right, moving on to the next. Yes, please. So enclosed within the integuments is a mass of cells known as nucellus. So what is the ploidy of nucellus here? The ploidy of ovule, the integuments or nucellus or any cell till now is 2n. Nucellus, what is the ploidy of each and every nucellar cell? 2n. Please note down these things, right? Cells of nucellus have abundant reserve food material. What does this indicate? That it is going to be a nutritive tissue itself. So nucellus is embryos. Located in the nucellus is the embryo sac, which will form later on, right? An ovule generally has a single embryo sac. If there are more than one, then it will lead to polyembryony stage as well. Now, microsporogenesis. Now, see, in case of anther, all the cells present within the anther were acting as sporogenous, means spore generating cells. Unlike anther, here only one cell in this nucellus will act as a megaspore mother cell, and preferably towards the micropyle. Okay, a nucellar cell, single nucellar cell towards the micropyle will act as a megaspore mother cell. Only one cell. Is that clear? And that is going to undergo meiosis now to form 4 megaspores. But unlike in microsporogenesis, here out of the 4 only 3 will degenerate and only 1 will remain functional. Okay, so see here, so formation of megaspore from megaspore mother cell is called megasporogenesis. What is the ploidy of megaspore mother cell? Same as nucellus, 2n, and it will undergo meiosis. So the ploidy of megaspore will be n. Is that clear? So see, these are all nucellar cells, 2n in nature, and then wait, this is all 2n, right? So even megaspore mother cell is also 2n, and now see what's happening. See, yeah, so this megaspore mother cell undergoes one meiosis to form 4 megaspores, and ultimately only 1 will remain functional, and that 1 will further undergo 3 mitosis to form an embryo sac. So this is how it happens. See, it forms this 2 megaspore dyad and megaspore tetrad, and out of this tetrad only 1 will remain functional. Yes, and the one which remain functional is towards the chalazal end. Is that clear? So one which remains functional is towards the chalazal end. See, they've written micropylar end this side. So one on this side, that will be the chalazal end, will remain functional. Now one functional cell will further undergo mitosis, and it will undergo 3 mitoses. Now this mitosis is such that only karyokinesis would occur. So nucleus is dividing 1 to 2, 2 to 4, 4 to 8. So how many mitoses have occurred? 3 mitoses, and this was a question in 2025. How many meiosis and mitosis take place in a megaspore mother cell to form a mature embryo sac? Answer is 1 meiosis and 3 mitoses. Is that clear? While in case of microgametogenesis from microspore mother cell, pollen mother cell, it was 1 meiosis and 2 mitoses. Please let me know if these things are clear to you, right? And after these 8 nuclei have been generated, now they are going to undergo rearrangement in such a way that 2 synergids and 1 egg cell will be formed towards the micropyle. See, it is written. Where is this? So 1 egg and 2 synergids together form the egg apparatus. Where are they formed? They are present towards the micropylar end. In the center we have the central cell with 2 polar nuclei, and towards the chalazal end, guys, we have 3 antipodals, which are again going to draw nourishment from the outside nucellus and provide it to the embryo sac. Is this clear? So ploidy, see, question around ploidy. That's why I'm asking you to remember what is the ploidy of each and every cell in this chapter. You have to know this. So ploidy based question was asked in 2023. So what is the ploidy of antipodals? N. Ploidy of each polar nuclei? N. What is the ploidy of synergids? N. These were the 3 things asked. Now they can ask for egg cell or any other thing. Yes or no? So please focus on the ploidy, rather make a list of all these ploidies. I'm going to upload the list of all the ploidies of each cell in my Telegram channel. Make sure that you join that. So this is a filiform apparatus, one of the favorite questions of NTA to ask, right? So filiform apparatus is present in the synergids and they secrete a chemical to attract the pollen tube. Is that clear? So it is going to be a chemotaxis movement. So question around the sequence of events leading to formation of female gametophyte. This was asked in 2023. So I've already described it in the diagram. Let's quickly read. So megaspore mother cell undergoes the meiotic division, formation of 4 megaspores, 3 degenerate, and only 1 which remain functional develops into the female gametophyte. And which is the female gametophyte? This is the female gametophyte. The embryo sac, the mature embryo sac, which has egg cell in it, which has female gamete in it, that embryo sac will be known as female gametocyte. Yes or no? So pollen grain represents the male gametophyte and embryo sac represents, mature embryo sac represents the female gametophyte. Remember these lines. This was asked in 2023 as well as which is the female gametophyte was asked in 2024. Then see, functional megaspore divides mitotically how many? 3 times. This is the sequence going on. And then 5th was after meiosis, we know that 8 nuclei will be formed. So 6 out of 8 nuclei are surrounded by cell walls and 3 cells are grouped into at the micropilar end to form the egg apparatus. Egg apparatus consists of 2 synergids and 1 egg cell. This was separately asked in 2025, 2022. So this whole paragraph and clear understanding of which cell is becoming what gradually, what is the fate of every cell, that should be very, very clear. So in case there is any doubt, guys, please watch the marathon at a higher speed, right? But if there is no doubt at all, then let's continue here. Now the 3 cells at the chalazal end, they will be known as antipodals. So a typical embryo sac, see it was asked twice, 25 as well and 21. A typical angiosperm embryo sac at maturity has 8 nuclei and 7 cells. Why 7 cells? Because 2 nuclei are present in the central cell itself. Is that clear to all? So this paragraph is one of the very important paragraphs. Then coming on to pollination, where the pollen grains are going to transfer from anther to the stigma. So if it occurs within the same flower, then this is known as autogamy, which is true self-pollination, right? For this pollination to occur, autogamy to occur, normally so anther and stigma should be exposed, well exposed. Complete autogamy is rather rare. Remember this line, complete autogamy is rather rare. So these are the lines to be marked. Complete autogamy is rather rare. In such flowers, so what are the conditions for complete autogamy? In such flowers requires synchrony in the pollen release and stigma receptivity. Second, the anther and stigma should lie close to each other. We have already discussed this. This is the second device, and we can call these devices as inbreeding devices. These are the inbreeding devices, because all the devices, all the things which help in ensuring or which ensure self-pollination are called inbreeding devices. So just like we have outbreeding devices which promote cross-pollination and prevent self-pollination, we have inbreeding which promote self-pollination and prevent cross-pollination. Is that clear? Now see, there are some plants, guys, some plants like viola, common pansy, oxalis and commelina, which produce 2 types of flowers, chasmogamous and cleistogamous. Remember, cle stands for closed, right? These are closed flowers. They will never open, right? But chasmogamous are typical normal flowers which are open around, right? So they produce both type of flowers. Now there is this line written here. See, the cleistogamous flowers are invariably autogamous, as there is no chance of cross-pollination landing on the stigma. This is 2022 question. See here. So cleistogamous flowers produce assured seed set. Very important line. Why? Because it is 100% sure that pollination will occur and the right type of pollen grains will fall and seed formation will occur. So it ensures the assured seed set even in the absence of pollinators. So do you think cleistogamy is advantageous? It has both advantages as well as disadvantages. So if we keep on performing, or if the self-pollination goes on happening one generation after other, then it will be disadvantageous as well. It has both the aspects. Is that clear? Now talking about geitonogamy, the definition, it was asked in 2023. What is geitonogamy? Transfer of pollen grains from anther to the stigma of another flower but same plant. Another flower but present on the same plant. And then xenogamy definition was asked in 2021. Can we expect the definition of even autogamy? Yes. So definitions are important as well. Anther to the stigma of a different plant. So which brings genetically different type of pollination. Is that clear? Now see, about geitonogamy, remember that functionally it is cross pollination, but genetically it is similar to autogamy. Is that clear? So genetically, because it is occurring within the same plant, it is self-pollination. Please tell me these points are clear to all. Yes, guys. Please tell me these points are clear to everyone. Right? Now, moving on to the agents of pollination. In both wind and water pollination, to compensate for the uncertainty, loss of pollen grains, flowers produce enormous amount of pollen grains. So maximum number of pollen grains are produced when the pollinators are abiotic in nature, that is wind and water. This was 2025 statement. Now coming here, pollination by wind is more common as compared to water. So amongst the abiotic ones, there are just two abiotic pollinators, wind and water. Which is more common? It is wind. And if we compare abiotic and biotic, then remember, then biotic takes the lead. Biotic pollination is more in nature as compared to abiotic pollination. Is that clear? Now see, what are the conditions for the wind pollination? The pollen grains have to be non-sticky, they have to be light so that they are well transported. They often well exposed stamens are present, and large, often feathery stigma to trap the airborne pollens. And remember this line also, they often have a single ovule in each ovary and numerous flowers packed into inflorescence. So these are the conditions which, or these are the adaptations for wind pollination, also known as anemophily. Please tell me these points are clear to you all. Please tell me yes. Is that clear to everyone? Yes, guys. Is that clear to everyone? So all the serious students in the class who are not chatting, but please take a second and give me a thumbs up. Let me know that everything so far is clear and these important lines are helping you to revise things in a better way. Yes, guys. Time for all the serious students to give me a thumbs up quickly. Right? Perfect. Let's move on. Let's come back. So this question was asked, see figure 1.10. This was 2024 question. They have given this figure in exam and asked, what does this figure represent? So guys, I am going to take a separate session, as I already told you, on diagrams, where we are going to understand what's written here, what are the implications, what are the different types of questions that can come up from each and every diagram. But this is an example of 2024 question. They've given this diagram and asked, given 4 different statements. So which of the following is the correct explanation of the given diagram? So it is a wind pollinated plant showing a compact inflorescence and well exposed stigma. So see, this is the type of question NTA is giving. So focus everywhere. Actually, it is not wrong. You want to be a doctor, you have to be very observant. You have to give importance to all, even the small details. You have to be detail oriented, right? So this is what NTA or say examination system is checking by giving such questions, right? So continuing further, pollination by water is quite rare in flowering plants. This was 2022 line, and limited to only 30 genera, and out of these 30 genera, mostly are monocots. Is that clear? Now see, apart from these higher plants, there are lower plant groups also, like algae, bryophytes, pteridophytes, which show pollination by water. We already know that, see, algae is aquatic 100%, bryophytes are near water, they are amphibians, the life cycle depends on water, and even pteridophytes, although have become terrestrial, true terrestrial, first terrestrial plants, but still have some dependence on water for sexual reproduction, right? So they, all these lower plants also need water, so they use water pollination. They use water as a pollinating agent. Is that clear? So some examples of water pollinated plants is Vallisneria hydrilla. So they will be not colorful. They do not produce nectar. So this was asked in 2024. Zostera is also water pollinated, but it is marine. These are freshwater. These are freshwater plants, while Zostera is marine in nature. Now, not all aquatic plants use water for pollination. In fact, most of the aquatic plants are also not water pollinated. Most of the aquatic plants will use other agents for pollination, maybe wind or insects, right? For example, water hyacinth and water lily, they are aquatic plants, but they are pollinated by insect and wind. This was asked in 2016 as well as in 2024. 2016 paper was really good paper. Now see, coming on to the next, these are the lines. So pollen grains in such species, see, they are talking about water pollinated plants like sea grasses. Here flowers will remain submerged in water. So pollens are not released on the surface but remain inside the water. In such species, the pollen grains are long, ribble-like, carried passively inside the water. Some of them reach the stigma and achieve pollination. Most of the water pollinated species, pollen grains are protected from wetting by mucilaginous covering. 2024, 2025, very recent years. Both wind and water pollinated flowers are not colorful and do not produce nectar. So in 2024, they have asked this question, that hydrilla and Vallisneria will produce nectar or will they not? Like similar question. And we are practicing these questions in special classes. Now, coming here to the biotic agents. So what are the biotic agents here? Bees, butterflies, flies, beetles, wasps, moths, all these birds. Which are the birds mentioned? Sunbirds and hummingbirds. They are the common pollinating agents. And now we are discussing even bats. These are the biotic pollinating agents. Is it clear? Now, out of this, guys, bees are the dominant biotic pollinating agents. This was asked in 2022. Now, even reptiles, gecko lizard and garden lizard have been reported as a pollinator. So now, in 16, they've asked the examples of reptiles. Can they ask examples of primates, the large animals like lemurs, arboreal tree-dwelling rodents? These are also expected questions, yes or no? Among birds, it is sunbirds and hummingbirds, which are very common pollinating agents. Is that clear? Now, majority of insect pollinated flowers are large, colorful, fragrant, and rich in nectar. This was asked in 2023. See here, the flowers pollinated by flies and beetles secrete foul odor, right? So they produce foul odor, but flies and beetles are attracted to that foul odor. This was also asked in 2022. Is that clear? Now, remember that nectar and pollen are the usual floral rewards. So you have to give them something in reward, right, for doing their work. So basically, plants and these insects and all these pollinators, they show a very close relationship, that is symbiotic relationship. Clear? They are dependent on each other. Now, other than nectar and pollen grains, there is another floral reward which is given in certain cases, and that is providing safe egg laying sites. This was asked in 2024. Now, one such example of a flower which provides safe egg laying site is Amorphophallus. This is the tallest flower known, 6 feet tall. Even I can fit into it. Anyways, so there are other examples like species of moth and the plant yucca. Again, they are symbiotically attached, associated with each other. They cannot complete their life cycle without each other. Is that clear to all? Is that clear to all? Right? So now moving on to outbreeding devices. What are outbreeding devices? These are going to promote, see, they promote xenogamy, that is cross pollination. Why? Because it is written clearly, continued self-pollination results in inbreeding depression, right? So this is the disadvantage of self-pollination. So first inbreeding device is that pollen grain, pollen release and stigma receptivity not synchronized. Is that clear? Second, so anther and stigma are placed at different positions. Is that clear? Third device is the self-incompatibility, and this is a genetic device. And the fourth one is production of unicellular flowers. Now, unicellular flowers, but if on the same plant, then it will promote geitonogamy, but it will not prevent self-pollination completely. Now, in order to prevent autogamy, so this will prevent autogamy but not geitonogamy. Now, this happens in case of castor and maize. The examples are important. While if you want to prevent self-pollination, that is both autogamy and geitonogamy completely, then in that case, they should be present on different plants. This is called diaceae. This is called diaceae. Is that clear? This is the fifth one. Example is papaya. So this will prevent both autogamy and geitonogamy. Please tell me these points are clear to all. Yes, guys. Please tell me these points are clear to everyone. Then pollen-pistil interaction. Now, pollen-pistil interaction is a chemical phenomena. See here. Yes. So, if a pollen grain is of the wrong type, the pistil rejects it. Pollen germination on stigma or the pollen tube growth in the style is a result of continuous dialogue mediated by chemical components of pollen interacting with those of pistil. This line was asked in 2016. Is that clear? Now, moving on here. So, if you want to study the pollen tube germination, you can use these plants like pea, chickpea, crotalaria, balsam and vinca, and take 10% sugar solution and let it remain in 10% sugar solution to draw nourishment, and after 15 to 30 minutes you will be able to observe, even under low power microscope, you will be able to observe pollen tubes coming out of it. Now, next point is artificial hybridization, very important step towards crop improvement program. So what are the steps in artificial hybridization? Number one step is emasculation. So emasculation is removal of anther. Emasculation is removal of anther, but this is done only in case of bisexual flower. Is that clear? If the female bears bisexual flowers only, then emasculation is performed. So this step is skipped or avoided if the flower is already a unisexual flower, because you are choosing a female flower. There is no anther itself already. So what would you emasculate, right? So this step is skipped in case the flower is female or unisexual. Now, emasculated flowers have to be covered with bag to prevent the contamination of its stigma with the unwanted flowers. This is why you do bagging, and this was question of 2022. After bagging, once it is receptive, once the stigma becomes receptive, pollen grains collected will be dusted onto the stigma and the flowers are rebagged and the fruit is allowed to develop. Now see, it is written here, if the female flower produces unisexual flower, there is no need for emasculation. So this is the step that has to be skipped. Now, talking about double fertilization, it is a characteristic of angiosperms only. Otherwise, there is single fertilization. But in angiosperms, there is double fertilization. It is 2017. Now, one of the two gametes move towards the egg cell, right? And this process is called syngamy. This is the fate. So, in 2019, this question was asked, what is the fate of the male gametes? So, one of them will move towards egg cell and fuse to form zygote. This fusion is called syngamy, while the other male gamete moves towards the two polar nuclei and fuse with it to form triploid pen. Is that clear? And because there are two fusions occurring, that is why it is known as double fertilization. And this fusion between two polar nuclei and one male gamete, this is known as triple fusion. Is that clear? Now, the central cell after triple fusion becomes the primary endosperm cell. Central cell will become PEC, primary endosperm cell, and the polar nuclei will become pen, primary endosperm nucleus, and the endosperm, while the zygote develops into the embryo. So, are you clear with this? Again, I'm mentioning the ploidy is very, very important. So, ploidy of these cells should be clear, guys. So, synergids are haploid, zygote is 2N, primary endosperm nucleus, pen, this is 3N in nature, zygote is 2N. So, all these highlighted things have been already asked in 2023. Is that clear? Now, look at the sequence of embryonic development. NTA love sequences. See the sequence here. So first of all, the zygote is formed by the fusion of male gamete with the egg cell. So first cell with which new life begins, zygote, it is 2N. Then zygote will form 2-cell stage. Then it will give rise to pro-embryo. After pro-embryo, it will form this globular embryo. After globular embryo, it will form this heart-shaped embryo. And after heart-shaped, it will give rise to the mature embryo. So you must know the sequence of this embryonic stages. So zygote, 2-cell stage, giving rise to pro-embryo. Then from pro-embryo forms. So 2-cell stage gives rise to pro-embryo to globular, globular to heart-shaped to mature. So this is the correct sequence of embryonic development. Is that clear? Now coming on to post-fertilization events. So we know that endosperm, see this line, endosperm development will occur before the embryonic development. Is it clear? And it is full of, the cells of this tissue is full of reserve food material. What is the ploidy of each and every cell of endosperm? It is 3N. Every cell in endosperm is 3N. It is a triploid tissue in case of angiosperms. And please let me know what is the ploidy of endosperm in case of gymnosperms. We have discussed that already. Quickly let me know what is the ploidy of endosperm in case of gymnosperms, guys. Yes, please. All the serious students here. Very good, Chaturya. Very good. The ploidy of endosperm in gymnosperms is haploid, because in gymnosperms it is a pre-fertilization structure, not a post-fertilization structure. Very good, guys. So Dr. Armfred, please rectify yourself and get the concept right. In gymnosperm it is haploid, while in angiosperm our endosperm is triploid. Is that clear? Now the most common type of endosperm development in which pen undergoes, most common type, undergoes nuclear divisions to give rise to free nuclei, that is free nuclear endosperm. And after that cell wall formation will occur to give rise to cellular endosperm. Is that clear? Becomes cellular. Now coconut water from the tender coconut, this was 2016. This is nothing but free nuclear endosperm. This is nothing but free nuclear endosperm, made up of thousands of nuclei, and surrounding white kernel represents the cellular endosperm. Is that clear? Now see, endosperm may be completely consumed or can be persistent. So pea groundnut beans, these are all dicots, and we have already read that in dicots they are completely consumed. So the dicot seeds are always non-endospermic seeds, with few exception. Let me know the exception in the chat, guys, or in the comment section. Name the dicot. We have discussed this in the previous session in morpho. Which dicot seed is endospermic in nature? Let me know. And similarly in castor and coconut, now these are monocots. These are monocots. Which monocot? Now monocots are generally endospermic. Again we have an exception here. Name the monocot which is non-endospermic. Is that clear? So you can easily answer these questions. Now the embryonic development, I have already told you what are the different stages. So zygote gives rise to, see, zygote gives rise to pro-embryo, then globular, heart-shaped and mature embryo. This is already discussed. Now a typical dicot embryo has embryonal axis and 2 cotyledons, right? And the portion above the level of cotyledons is epicotyle, which later forms the plumule, and the part below the cotyledons is called hypocotyle, which later forms the radicle or the root tip, and it is covered by root cap. Now in grass family, whenever they are saying grass, it means in monocots. Grass represents monocot. There is only one cotyledon. There is only one cotyledon situated towards the side of the embryonal axis. Is that clear? So see here the cotyledon is lateral to the embryonal axis, and other than that position of embryonal axis, another difference between dicot and monocot is presence of coleorhiza and coleoptile. These are some hollow foliar structure means some leaf like foliar structures means they are sheath like structures. They're going to cover, coleorhiza covers the radicle and coleoptile is going to cover the plumule. Is that clear? Now, talking about seeds, already discussed non-albuminous or X-albuminous, also known as a non-endospermic. So, no residual endosperm. That means endosperm completely used. What is the example? Majorly dicots. You have to give me the exceptional case here. And then albuminous seeds, or we call them endospermic seeds, where endosperm is left behind, that is C. These are all monocots. Right? Now, other than endosperm, there is another term called perisperm. See, the question has already been asked around perisperm. So, nucellus present around the embryo sac, right? That nucellus is also consumed generally, but in some cases there can be some nucellus left behind. So the residual or the persistent nucellus is known as perisperm. What is the example here? And that persistent nucellus is known as perisperm. See the examples: black pepper, beetroot, called beet. They have remnants of nucellus. Very important examples here. And then just like okay, let's talk about this. So a seed has to undergo this very important stage called dormancy. It may enter a stage called inactivity or dormancy, during which 10 to 15% moisture is left behind, rest all is gone. And if the favorable conditions remain, it will germinate, otherwise it will go in a state of dormancy. Is that clear? Now the wall of ovary will become the wall of fruit. These are post-fertilization events. Now, the wall of fruit is known as pericarp, and we have already discussed that pericarp can be differentiated into 3 layers, that is epicarp, mesocarp, and endocarp. Is that clear? And we have discussed the nature of epicarp, mesocarp, and endocarp in case of drupe fruit. The example of drupe is mango and coconut. We have discussed that. So these can be differentiated here. Now, similarly, fruits can be fleshy like in guava, orange, and mango, or it can be dry as in case of mustard and groundnut. Is that clear to all? Now, false fruits also we know. In false fruits, usually the thalamus contributes to the fruit formation, and in case of true fruits, it is only the ovary that results. What is the example of false fruits? Strawberry, cashew, and apple. True fruits there are so many: mango, orange, these are all true fruits, right? Now, talking about the fruits which develop without fertilization, parthenocarpic, and hence they are seedless also. For example, banana. Now, this parthenocarpy can be induced also by the application of growth hormones like auxins. We induce parthenocarpy in tomato. We have read the chapter in plant growth and hormones. Is that clear? Now, so female gamete into embryo. If female gamete changes into embryo without fertilization, this is parthenocarpy. This is asked in 2019. Now there are some examples of very extended seed dormancy. See here, the oldest case of lupine or Lupinus articus from Arctic tundra. It showed dormancy of 10,000 years. That means after 10,000 years, when the seed was excavated, it germinated to form a new plant. And then another case of 2,000 years old viable seed of date palm, khajur, Phoenix dactylifera from King Herod's place. Can you get these questions and match the following? Yes, why not? So please be prepared with these type of questions as well. Is that clear? Now, talking about apomixes, it is to produce seeds without fertilization. Is that clear? Production of seeds without fertilization. No fertilization means it is asexual type of reproduction, but still producing seeds means it is mimicking sexual reproduction. So, apomixes is a form of asexual reproduction that mimics sexual reproduction. This is important. So, here diploid egg cell is formed without reduction division and develops into embryo without fertilization. Examples are Asteraceae and grasses. These are the examples of apomixes, and it was asked in 2016. Then we have polyembryony. So, what is polyembryony? Occurrence of more than one embryo in a seed. This is called polyembryony. The example is citrus, not all, in many citrus and mango varieties. How does this happen? Nucellar cells surrounding the embryo sac also start dividing, protrude into the embryo sac and develop into embryos. So because ovule is the same, so seed will be one, but that seed will contain more than one embryo. Is that clear? One seed with many embryos, that is polyembryony. Please tell me these points are clear to you all. And what is the advantage of this amphimixes, apomixes? What is the advantage here? So apomixes is a great technique. See here. So apomixes, because apomixes is, so seeds are produced, but without fertilization, there is no evolution here. So no new characters have been added, right? So basically we can produce apomictic seeds because there is no segregation of characters in the hybrid progeny. So that's where the apomictic seeds are very, very advantageous. So it helps in conservation of characters and also produce seeds. So you can produce seeds without the segregation of the characters. So that is a big, big advantage in the field of seed industry. Are these points clear to all? Please tell me. So with this, guys, we complete the first chapter, and now moving on to the next chapter. Dicot seeds, castor, monocot seeds, orchids. Right, Haswant? Very good, very good, very good. All right. Now, moving on to the next chapter of class 12, that is human reproduction. So see here, what are the events in the particular order? So the reproductive events in humans. So the reproductive events, the first being gametogenesis, right? Sperm production in male and ovum in female. Second is transfer of sperms into the female genital tract, that is by insemination. Then third is the fusion of male-female gamete, that is called fertilization. Fourth is the formation of zygote, and finally there is formation of embryo, followed by formation and development of blastocyst, and embryo will be implanted, that is sixth, and finally this gestation leading to delivery of the baby or parturition. So you should know these events in sequence. Now moving on to male reproductive part. So male reproductive part is located in which region? Pelvic region. What does it include? You already know. Where are testes located? Testes are present outside the abdominal cavity in the in the pouch called scrotum. What is the function of scrotum? To maintain low temperature. How low? 2 to 2.5 degrees Celsius lower. Why? Because this is necessary for spermatogenesis. Is that clear? Now, because we already know the process of spermatogenesis, first it is multiplication phase, which includes mitosis, then growth phase and maturation phase. Maturation phase involves meiosis. So exactly speaking, meiosis requires lower temperature. The maturation phase requires lower temperature. Is that clear? Now, what is the shape? It is oval, length 4 to 5 and 2 to 3 centimeter, covered by dense covering. How many compartments does testes have? 250 compartments, and these compartments are known as testicular lobule. Now, this is about each testes. Each will have 250 testicular lobules, and each lobule will have around 1 to 3 highly coiled seminiferous tubules. So seminiferous tubule is the unit of testes, right? And it is lined by, see, each seminiferous tubule is lined by two types of cells. One, the male germ cells known as spermatogonia, and second is the Sertoli cells. Is it clear? Now, male germ cells is going to undergo meiotic division to form sperms. We already know. And what is the function of Sertoli cells, also known as nurse cells, right? So Sertoli cells are called nurse cells because they provide nourishment. Sertoli is in the name of scientist, right? So they provide nourishment. Now, outside the seminiferous tubule lies the interstitial spaces filled with interstitial cells, also known as Leydig cells. Asked in 2020, what is the function of Leydig cells? They produce androgens. Is that clear? Now, other than Leydig cells, there are some immunologically competent cells also present in the interstitial space. Is that clear? Now, what are these accessory ducts like? So the question around pathway of sperms was asked in 2024 as well as 29. So seminiferous tubule, then in rete testes, then vasa frangia, then epididymis, then vas deferens, leading to ejaculatory duct and finally urethra. So it receives a duct from, it means vas deferens receives a duct from seminal vesicle, opens into the urethra as ejaculatory duct. What does it mean? Means vas deferens and duct from seminal vesicle will first form ejaculatory duct and then open into urethra. So this line as was directly asked in 2023. Is that clear to everyone? So do we see a convergence of excretory system and urinary system, excretory system and reproductive system here? Please tell me, guys. Do we see a convergence here? Yes, guys? Is that all clear? Now, moving on to the next part is, see, the male external genitalia is penis. And there is a special tissue, the erectile tissue, which helps in erection. And the enlarged part of penis is called glans penis, covered by a loose fold of skin known as prepuce or foreskin. Now, accessory glands. There are 3 glands: seminal vesicles, prostrate, and bulbourethral. Which of them are single or paired? Bulbourethral and seminal vesicles are paired, while prostrate gland is single. Is that clear? So, secretions. Now, note this line. Secretions from these glands. So, all the 3 glands will secrete to constitute seminal plasma. So, the constitution or components of seminal plasma comes from all the 3 glands. And what are they rich in? What is the composition? Fructose, calcium, and certain enzymes. You don't have to learn a lot. Fructose, calcium, and certain enzymes. And let me tell you, fructose is present only in semen. There is no other body fluid which contains fructose. Is that clear? So, secretions of only bulbourethral gland, to be particular, is helpful in lubrication of penis. This was asked in 2023. Yes. So, can we expect the question around seminal plasma in 2026 Re-NEET exam at least? Yes, we can. So one thing is for sure, guys, they are not going to ask you anything out of NCERT. Just take it for granted. NTA is not going to do that with you now. At least not at this point. Everything will be asked from NCERT. Direct questions will be asked as well. And match the following will be done. And we are practicing these questions already. So don't think that what will be the level of Re-NEET exam. It will be same as it was in NEET exam 2026. I really don't think so that they are going to ask something out of the NCERT just to set a new standard here. No, this is not the time for NTA to set new standards. The new standards will be set only in conduction of fair and secure exam to earn your trust again. That's the best NTA can do. So don't worry about anything else. So what will be the level? It will be the same level. Not very difficult, not very easy. Because earlier also, whenever, if we search history of NTA and the NEET exam, so whenever there was a Re-NEET conducted, the level was almost the same. And also we have seen some repetition of the questions. So please make sure that you revise your NEET exam also. So it is a great opportunity for you. Okay, so stop looking at the negative side. Let's be positive now. NTA is okay, trying to gain trust. Let's give it another chance because we don't also have any other alternative. It is our career also. So give it another chance, be positive, revise your NEET 2026 paper. Things may be repeated. Clear? All right. Now, moving on to female reproductive system. So pair of ovaries, uterus, oviduct, cervix, vagina, and external genitalia located in the pelvic region. And mammary glands also a part of this system, integrated structurally and functionally to support the process of this. So although mammary glands are modified sweat glands, part of your skin, but still studied along with the reproductive system. Why? Because they're structurally and functionally related to support all the reproductive activities. Is that clear? Now, each ovary is covered by epithelium which encloses ovarian stroma, and stroma is divided into outer cortex and inner medulla. Is that clear? See the structure and let's, okay, see here. The oviducts or the fallopian tubes, uterus, vagina constitute the accessory ducts. What are the accessory ducts? Fallopian tubes, uterus and vagina. 1, 2, 3. Now, how long are these fallopian tubes? 10 to 12 centimeter long from periphery of each ovary to the uterus. Now, the part of the ovary towards the, part of the fallopian tube towards the ovary. So, this is ovary, part of fallopian tube towards the ovary is infundibulum. Now, the parts were also asked in 2024. and you can be asked to answer or tell the sequence of fallopian tubes from ovary towards uterus or the vice versa. So if you go from ovary towards uterus, the answer is infundibulum, then ampulla, and then isthmus. So read the question, what are they saying? From ovary towards uterus or from uterus towards ovary? So answer will reverse according to that. So funnel shaped infundibulum, right? It is a part closer to the ovary and it possesses these finger-like projections known as fimbriae. What is the function of fimbriae? This was also asked, to collect the ovum after ovulation. Is that clear? So these are finger-like projections, fimbriae, lying just above the ovary. They will pick up the ovum after ovulation. Is that clear? Now moving on here, the part. Sorry. Yes, infundibulum leads to a wider part known as ampulla, and this is the place for fertilization. This is the site for fertilization. Now the last part of oviduct, which is narrow. So ampulla means dilation. So ampulla is wider, isthmus is going to be narrow, and it joins the uterus. Last part of oviduct and near the uterus, so joins to the uterus. So uterus is single, also known as womb, also known as hystera. It is inverted pear shaped, supported by ligaments attached to the pelvic, opens into the vagina through a narrow cervix. Is that clear? Now the cavity of cervix, see, cervix is the posterior part of uterus itself, right? So the cavity of cervix is called the cervical canal. Along with the vaginal canal, it is known as the birth canal. This was asked in 2023. Just the composition of birth canal. Composition of birth canal. Is that clear? Please tell me these points are clear. Right? And now, the wall of uterus has how many layers? 3 layers. External thin membranous perimetrium. Outside to inside. Perimetrium, myometrium, and inner is endometrium. So name itself helps you out. You don't have to put in more efforts. Peri means outside, boundary. Then myo means the muscle. So the one made up of smooth muscles. What type of muscles are there? Smooth. They are not in your control. And inner layer is glandular because it is going to secrete hormones as well. Estrogen, the second peak during the menstrual cycle, right? That comes from endometrium. So lines the endometrium. So endometrium shows cyclic changes. So what is the importance of myometrium? It shows cramps. Even during periods, cramps or the strong labor contractions, they all occur in myometrium, right? And endometrium holds the embryo. So implantation occurs in the endometrium, and every month it undergoes the cyclic changes. So myometrium, strong contractions for the delivery. So basically oxytocin, the birth hormone, is going to act on myometrium. Clear? Now the female external genitalia, see, female external genitalia known as vulva, includes mons pubis, labia minora. This whole thing was asked in 2024 in the form of match the following. So what is mons pubis? Cushion of fatty tissue under the skin and pubic hair. What about labia majora? Fleshy folds of skin surrounds the vaginal opening. What about labia minora? Opening of vagina is it is a paired fold under the labia minora, and the opening of vagina is covered by a vagina is often covered partially by a membrane called hymen. What about clitoris? It is a tiny finger-like structure, lies at the upper junction of two labia minora. Remember these things. So this whole paragraph was asked in the form of match the following. Now, next, again in 2024, hymen is often asked during the first coitus, torn during the first coitus. However, it can also be broken by a sudden jolt or a fall, and insertion of vaginal tampon, and active participation in some sports like horseback riding, cycling, etc. Therefore, presence or absence of hymen is not a reliable indicator of virginity or sexual experience. All these lines, this whole paragraph was asked in 2024. See the importance. Clear? Now, coming on to mammary gland. So, the question was asked about components of mammary gland from proximal to distal. See, this is how they ask. Proximal to distal. Which is the proximal end here? Proximal means near to the body. So, this must be the proximal end. and this must be the distal end away from the body. Yes, so proximal to distal, so you have to answer in this order, starting from the mammary lobe. Here, mammary lobes which contain alveoli and secrete milk. So from alveoli, milk goes into the mammary tubules, then into the mammary ducts, then into the mammary ampulla where it is stored, and finally when it has to be released, it moves out through lactiferous ducts and opens at the nipple. So this is how it is asked, from proximal to distal. This was in 2022. So the same pattern can be followed in asking some other questions also. Just remember, proximal means near, distal means far, distant. So proximal is more towards your body, distal means away from your body. Can you do such questions? Now, talking about gametogenesis, see, it was asked here, how many the secondary spermatocytes undergo second meiotic division to form 4 equal haploid spermatids. The spermatids are transformed into spermatozoa and the process is called spermiogenesis, as twice 24 as well as 18. Now sperms are finally released from seminiferous tubules by the process of release of sperms from seminiferous tubule is called spermiation. Is it clear? Now, sequence of events, again, sequence are important. So, the hormonal regulation in sequence. Gonadotropin hormone, releasing hormone coming from hypothalamus. So, hypothalamus releases GnRH, which impacts the anterior pituitary to release gonadotropins, which are of two types, LH and FSH. So, LH will act on Leydig cells in case of males, and Leydig cells will stimulate the synthesis of androgens, while androgens will stimulate the process of spermatogenesis, that is testosterone. Now, FSH acts on the Sertoli cells and stimulate the secretion of some factors which help in the process of spermiogenesis. This whole thing was asked as a sequence in 2023, and the hormonal regulation in males in leaving the blanks for the hormonal regulation was asked in 2024 and 16. So just pay attention here. FSH acts on Sertoli cells, and what do Sertoli cells do? They stimulate the secretion of factors for the process of spermiogenesis. Is that clear? It is spermiogenesis, the last step, that is the conversion of spermatids into sperms is spermiogenesis. Clear? Now again, functions in match the following 2025. Head contains what? Haploid nucleus. Acrosome present in the head is filled with enzymes. What about middle piece? It provides energy because it contains a lot of mitochondria. Then the tail is meant to facilitate the sperm motility. Clear to all? Now, the seminal plasma along with the sperms forms the semen. So semen is a term used for seminal plasma plus sperms, and you have read it just now, that what is seminal plasma composed of? Fructose, calcium and certain enzymes. And which 3 glands are contributing for the formation of seminal plasma? All the 3. Is that clear? Now see, the functions are maintained by testicular hormones, that is androgens. Again, this whole paragraph is important, but oogenesis is initiated during the embryonic development stage when couple of million of gametes, mother cells, oogonia formed within each ovary. No more oogonia are formed and added after birth. So this was 2025, 2022. The whole paragraph is important. So when we are born, these oocytes, they are arrested in prophase 1. And which stage? Diploteny stage, and they are called primary oocytes. Each primary oocyte is surrounded by a layer of granulosa cells to form primary follicles. Many primary follicles degenerate during this phase from birth to puberty, and at puberty we are only left with 60,000 to 80,000 primary follicles with primary oocytes. The primary follicles get surrounded by more layers of granulosa cell and a new theca and is known as secondary follicle. This was asked in 2023. So the development of follicles is very, very important. So what is the difference between primary follicle and secondary follicle, from tertiary and graphene follicle? You must know these differences very clearly and discreetly, right? Now, so follicle soon transforms into tertiary follicle. What is tertiary follicle like? Which is characterized by a fluid filled cavity called antrum. So tertiary follicle also has an antrum. but a smaller one. But later on, the theca cells are layer is organized into interna and externa. And once all this is formed, and also the primary oocyte completes its first meiosis and an unequal division, resulting in formation of a large haploid secondary oocyte and a tiny first polar body. Is that clear? So these are important. Secondary oocyte contains bulk of nutrients with rich cytoplasm of the primary oocyte. Clear to all? Now after this, when the first meiosis is done, it is secondary follicle and primary oocyte. Now that stage is known as graphene follicle or the mature follicle. Now secondary oocyte when forms, it releases a membrane called zona pellucida. The graphene follicle now ruptures to release the secondary oocyte ovum from the ovary by the process called ovulation. Now see, they have rectified this in new NCERT. Earlier it used to be ovum written here. So now we know it is not the ovum which is released during ovulation, it is the secondary oocyte. Yes or no? Yes guys. So is this clear to all? Please tell me, are these points clear? Yes, all the serious students here. I want all the serious students. Take a second and give me the answer. All clear? If you want any other help, if you want to ask something, this is the time. Because I don't check the chat usually. When I'm asking serious students, I am actually looking into the chat. Please let me know. Yes, guys. All clear? They gave extra time and rough pages. Will this Re-NEET be tough? Haripriya, I've already answered your question. I don't know. Were you not listening to me 5 minutes back? You should be. So yes, and NTA is trying to gain your trust. Give them another chance because we also don't have any other alternative, right? They are giving you another chance. They're trying to earn your respect and your trust in NTA again. Let's give them one more chance because we don't have any other alternative and it's about our career also, right? So yes, they are giving you extra 15 minutes to fulfill all those exam those things, all the formalities here. So they're giving you extra 15 minutes. They are giving you extra page for doing the rough work. Do as much rough work, draw as many diagrams as you want, but do it judiciously. Extra 2 pages doesn't mean that you have to do unnecessary work there. Learn the tricks. If there are tricks, learn the tricks because you have to save time. And please guys, don't get befooled by these extra 15 minutes. So, okay, now I have extra 15 minutes. I don't have to manage my time. This is not the case. It's just NTA trying to earn your trust. Next year onwards also, I don't think there will be extra 15 minutes. If there is, fine. If there is not, don't get befooled by that. Okay. So don't practice questions and don't practice your paper in such a way that you have extra 15 minutes. Practice the paper with the same speed. Manage your time like you have only 2 hours 45 minutes, right? Because earlier also we were trying to practice things in a similar way. We were trying to manage time in a similar way so that we can take out 15 minutes for the exam things to complete. So practice your paper in a similar way. Don't, count these 15 minutes for completing your paper. It is for completing the formalities. Is that clear? What is the difference between, okay, Venkatesh is asking, what is the difference between gametophyte and gamete? See, gamete is that cell which will undergo fertilization, right? Male gamete, sperm, female gamete, egg. Gametophyte is the structure which contains gamete. If we talk about plants, then pollen grain is the gametophyte which contains 2 male gametes. In case of female, then it is an embryo sac which contains the egg cell. So gametophyte means which contains gamete or which bears gamete. Is that clear? I hope this point is clear to you. Is that clear? Right? All right, now moving on to the next. So Graafian follicle, guys, now ruptures to release secondary oocyte and from the ovary by the process of ovulation. Major differences between spermatogenesis and oogenesis should be very clear in your head, because there was a question based on differences between two in 2022. I'll be practicing these questions in special classes. Now, numerical based question was also there in 2022. It can come back in 2026. So what was this? So see, we know that sperm production occurs continuously. This was asked, this was a difference based question. So we know that oogenesis is a continuous process of around 60 to 70 days, while spermatogenesis, I'm so sorry. So spermatogenesis is a continuous process, right, of 60 to 70 days, while oogenesis is a discontinuous process. Starts in the fetal stage, then pauses for around 11 years till you reach the puberty, then restarts after puberty, and then ceases completely at menopause. So it is a process which occur in spurts, right? So this is oogenesis, this is spermatogenesis. We know at the end of spermatogenesis we'll have 4 sperms from 1 spermatogonia, while here 1 oogonia is going to give you 1 ovum. So the ratio is 1:4, and here the ratio is 1:1. So if you want 1000 sperms, you need 250 spermatogonia, 250 sperm primary spermatocytes, 500 secondary spermatocytes, and 1000 spermatids, because spermatids and number of sperms is equal. Is that clear? While if you need 1000 ova, you need 1000 oogonia also. Please see. Are these points clear? So this was asked in 22, and even the process has been asked in 2025. Now, reproductive cycle in non-primate females is known as estrous cycle. This was asked in 2023, guys. But in case of primate females, it is known as menstrual cycle. First menstruation begins at puberty and known as menarche. It was asked in 23, 25 as well. And then there were so many questions from this graph. In 2018, you must know all the phases in correct sequence. Then negative feedback of ovarian hormones on pituitary hormones, asked in 2016. Is that clear? So breakdown of endometrium is known as menses and leads to the menstrual phase. This is asked in 2018. So all these questions are important, sequences, everything. Now, very important line, lack of menstruation may be indicative of pregnancy. Answer is, the statement has word may be, not always, because lack of menstruation can also indicate hormonal imbalance, like in PCOS, right? So it is not always indicative of pregnancy, it may be indicative of pregnancy. That was asked in 2023. So if they say it is always indicative, answer will be false. Is that clear? Now coming on to, yes, hormonal regulation in females. Hormonal regulation. See, LH and FSH increases gradually during the follicular phase. Asked thrice. 16, 22, 24. Then FSH leads to follicular development as well as secretion of estrogen by the follicular growing follicles, right? Then in the middle of the cycle, about 14th day, there is rapid increase of LH leading to LH surge, which induces rupture of Graafian follicle. And what do you call this? This process of release of ovum is known as ovulation. So LH is responsible for ovulation. Asked 2016, 25, 24, 20, 4 times, right? Now see, the Graafian follicle transforms as corpus luteum. Corpus luteum secretes large amount of progesterone, which is essential for maintaining of endometrium. Endometrium is necessary for implantation of fertilized ovum. And which embryonic stage? Blastocyst. 23. Now during pregnancy, all the events of menstrual cycle stop. There is no menstruation in the absence of fertilization. If fertilization doesn't occur, then corpus luteum will degenerate. And in how many days? 14 days. This causes disintegration of the endometrium and leading to menstruation. Is that clear? Menstrual cycle ceases around 50 years of age. This is known as menopause. Cyclic menstruation is indicator of normal reproductive phase and extends between menarche and menopause. The whole line is important. If you have a regular cycle, that means you have a normal reproductive phase going on. And this extends between, normal cycle is between menarche and menopause. Asked twice again, repetitively, once in 23, then in 24. Can it appear in 26? Let's see. Now, Fertilization and implantation. The motile sperms swim rapidly, pass through the cervix, enter into the uterus, finally reach the ampullary region. See, within 24 hours it has to do this, because the viability of sperms after ejaculation is just 24 hours. Before ejaculation, it can be for a few weeks to months, but after ejaculation, it only has 24 hours to reach the egg. Is that clear? Or the secondary oocyte. Is that clear to all? So you should know the pathway after ejaculation. What is the pathway after ejaculation? That is vagina, then cervix, then uterus, then fallopian tube, and it has to end up in ampulla, where the secondary oocyte is waiting. So fertilization can occur only if ovum and sperms are transported simultaneously to the ampullary region of fallopian tube. Why simultaneously? Because of the viability. Because sperm has only 24 hours while ovum has 24 to 48 hours. But the sooner, the better it is, the more chances. Is that clear? Process of fusion of sperm within the ovum is fertilization. Sperm comes in contact with zona pellucida first, right, of the ovum. So all these questions, look at this, right, and induces the changes in the membrane and blocks the entry of additional sperm to prevent polyspermy. Thus, it ensures that only one sperm can fertilize an ovum. The secretions of acrosome help the sperm enter into the cytoplasm through the zona pellucida, and the sperm, so guys, the receptors for sperm binding in case of mammals lie on zona pellucida. Is that clear? Now this induces the completion of meiotic division of the secondary oocyte. Second meiotic division is also unequal, results in the formation of second polar body. Now this was asked in 2019 after sperm entry but before fertilization. So that's when second meiosis occurs. Is that clear? So when the sperm enters, it completes the meiosis 2. It waits for the oocyte to complete meiosis to form ovum, become ovum, and then it will fuse to form a zygote. Are these points clear? So we know about sex determination already. So the meiotic divisions, what type of divisions occur in the zygote? Meiotic division starts as it moves through the isthmus towards the oviduct, isthmus of the oviduct, and these meiotic divisions are known as cleavage. What is the difference? There is no growth phase here. All right. So these are the stages. So first it forms the morula. So after zygote, the prominent stage is morula, then morula into the blastocyst or blastula, and finally when it starts forming the germ layers and all, it becomes a gastrula. So blastula will have two layers, trophoblast and inner cell mass. Trophoblast will provide nutrition, and then inner cell mass gets differentiated into embryo. That is why inner cell mass is also known as embryonal mass. Is that clear? Now, uterine cells divide rapidly and covers the blastocyst. Blastocyst becomes embedded in the endometrium of the uterus. This is called implantation, and implantation is the beginning of pregnancy. Please tell me these points are clear. Yes, guys. So talking about pregnancy, so after implantation, finger-like projections appear on the trophoblast, which is known as chorionic villi, and surrounded by the uterine tissue and the maternal blood, and they will become interdigited, means they fix into each other like this tightly, jointly form a structural and functional unit between developing fetus and maternal body. This is called placenta. Very important. So the structural and functional unit between embryo and the mother, that is placenta. What are the functions? It acts as an endocrine tissue. What are the hormones? HCG, HPL, estrogen, progesterone. These are the questions asked. And towards the end of the pregnancy, there is another hormone called relaxin also secreted by ovary. But which part of ovary? Corpus luteum secretes it. Is that clear? So corpus luteum is maintained during the whole pregnancy, entire pregnancy. So during pregnancy, there are other hormones also. See, this was a question. This was a question also. HCG, HPL, relaxin. These are the hormones which will be produced only during the pregnancy, not otherwise, but only during pregnancy. This was asked during, this was asked in 2022. See here. Right? But there are other hormones like estrogen, progesterone, cortisol, prolactin, thyroxine. They are also increased in the maternal blood. But We see other hormones otherwise also in our body, but these three hormones, HCG, HPL, and relaxin, they are not produced otherwise in the non-pregnant stage, right? Now, increased production is essential for supporting the, why are they increased? To support the fetal growth, metabolic changes in the mother, and maintenance of pregnancy. So these are assertion and reason. Yes, these are assertion and reason. Do we see these things here? Immediately after implantation, the inner cell mass into the outer layer ectoderm and inner called endoderm. So the inner cell mass, the first two germ layers formed are ectoderm and endoderm. Mesoderm soon appears between ectoderm. So the last germ layer formed is, this is the last one to be formed. Last germ layer is mesoderm. Is that clear? And these three germ layers will give rise to all the tissues and organs. So when these germ layers appear, this is the beginning of gastrula stage. So this is what happens during gastrulation. Is that clear? Now, another very important paragraph already asked, 24, 25. Let's see, Re-NEET 26. So after 1 month, embryo's heart is formed. So which is the first organ to develop completely? That is heart. The first sign of growing fetus is the heart sound. By the end of 2nd month, limbs and digits. End of 12 weeks, that means 3 months or first trimester, most of the major organ systems. Then limbs and external genitalia also formed. Then first movements of the fetus, they occur during the 5th month, and appearance of hair on the head. Then by 24 weeks, that is 6 months, or the end of 2nd trimester, eyelids separate, eyelashes are formed, body covered with fine hair, and end of 9 months, ready for delivery, fetus fully developed. Are these points clear? Then parturition. What is parturition? Delivery of the baby or the childbirth. Complex neuroendocrine mechanism. This was asked in 2024. What the signals for the parturition are known as fetal ejection reflex. So where do the signals originate from? Originate from fully developed fetus, number 1, and second is placenta, right? And what does it trigger? It triggers the release of oxytocin from where? Maternal pituitary. From maternal pituitary. Is that clear? What is important for parturition? This was asked in 21. Release of oxytocin from maternal pituitary, synthesis of prostaglandins. These prostaglandins will stimulate further labor contractions, right? And increase in estrogen and progesterone. Now this is oxytocin will cause strong uterine contractions and release of milk. Last not the least, so production of milk starts. See, mammary glands undergo differentiation during pregnancy and starts producing milk towards the end of pregnancy. So before birth also milk production starts slightly, and towards the end of pregnancy, and this is called lactation. The milk produced during initial few days is called colostrum, which contains several antibodies important for disease resistance in the newborn baby, because proper immune system is not developed. So breastfeeding during initial period is recommended by doctors for bringing up a healthy baby. This is assertion and reason, right? Already asked in 2024. Please tell me that these points are clear to everyone. Yes, guys. Please tell me all these points are clear. So breastfeeding during initial period. Oh no, Hema, my voice is loud enough. Please check your headphones. Is that clear to all the serious students? Again, I want your attention here. All the serious students. Hi, Anuradha. Akshay, clear. Hema, can you stop spamming? Don't be an attention seeker, guys. Please. I read your messages after the end of every chapter. Is that clear? All right, yes. Let me go and get a sip of water. Please get ready with another chapter, and we are going to start and complete it. All right, guys. I am back. Yes, please. Can you clip the mic to your collar? I will do that. I have already done that. I just went for a sip of water. All right. Moving on to the next chapter or the last chapter of the first unit, that is reproductive health. Yes, guys. Yes, according to WHO, what is the definition of reproductive health? It is total well-being or in all aspects of reproduction. And what are the aspects? What are the aspects here? It is physical, emotional, behavioral, and social. There are 4 aspects that we have to discuss or consider. This definition was asked in 2023. So India was the first among the countries in the world to initiate the first country to initiate a national level program to attain reproductive health as a social goal, and these programs were termed as family planning, initiated in 1951. So India was the first country in the world. Wow. Improved programs and family planning was gradually named as, this was named as RCH, reproductive child health care, right? What is the objective of RCH? Different objectives. Number 1, creating awareness among the people about reproduction aspects and providing not only awareness, second is providing facilities and support for building the reproductively healthy society. So just awareness won't work. So provide awareness to the people and provide them facilities. These are the 2 major objectives of RCH. Is that clear? Then other things, audio, video, visual and print media government and NGOs creating awareness amongst people and students and all these people. Introduction of sex education at the right to provide the right information to the young people about and discourage children from believing in myths and misconceptions. Proper information about reproductive organs, adolescents and related changes, about hygienic sexual practices and STDs like AIDS, and then educating the people, especially fertile couples and those in the marriageable age, about birth control options, care of pregnant mother, postnatal care, importance of breastfeeding, and provide and equal opportunities for the male and female child, especially those who love having beta, right? What address importance of bringing up socially conscious healthy families, awareness of problems due to uncontrolled population growth, social evils like sex abuse, sex related crimes need to be created to enable the people to think and take up necessary steps to prevent them and thereby build up a socially responsible and healthy society. Also there was a question in 16 about amniocentesis. When is it done? Usually done between 14 to 16 weeks of pregnancy, and it is a prenatal sex determination test. But is this the objective? No, it is not the objective. But the objective, well, not in India because of female feticide. So in India, what is it meant for? This test is used for the presence to test the presence of certain genetic disorders like Down syndrome, hemophilia, sickle cell anemia, right? And to determine the survivability of the fetus, whether it will survive or not. So there is a statutory ban on amniocentesis for sex determination in India to legally check the increasing menace of female feticide. These are the questions, all questions. Now, also, can it be used to detect cleft palate? That was asked in 2016. No. Can be detected by sonography. So cleft palate is a structural deformity. It will be identified or detected by sonography. We don't need to conduct amniocentesis for that purpose. Now Saheli, a new oral contraceptive female, was developed by scientists at CDRI. So first of all, you must know the full form of CDRI, which is Central Drug Research Institute. Where is it? It is in Lucknow. And it was this institute, CDRI, which developed Saheli. Actual name of Saheli is centochromon. Why do we call Saheli? Because of its friendly nature towards a female. Very few side effects, that's why. What is the function or mode of action of Saheli? It blocks the estrogen receptors in uterus, preventing implantation. This was asked in 2018. Well, population stabilization, you must know this data for match the following aspect. So world population 2 billion in 1900. Okay, 6 billion by 2000, 7.2 billion in 2011. Is that clear? Then in India, see the trend in India, 350 million at the time of independence, that is 1947, 1 billion by 2000, crossed 1.2 billion by May 2011. So you should know this data for match the following. Now there is rapid decline in death rate, MMR and infant mortality rate, and increase in the number of people in reproducible age. This has led to increase in population. Why has the population shown such a rise? Because of these four aspects: decline in death rate, MMR, IMR, and increase in the number in reproducible age people. Is that clear? Now, the population growth has been less than 2% in 2011, a rate at which population could increase rapidly. Is it clear? Now, most important step to overcome the problem is to motivate small families, use of contraceptive methods, these posters like and slogans like Hum Do Hamare Do, we two are two, and many couples adopting one child norm, then raising of marriageable age, female it is 18 and males it is 21. Now there are proposals to increase them further, and incentives given to the couples with small families. Right now, important, ideal contraceptive. What are the features of ideal contraceptive? Number one, easy to use, easily available, effective and reversible with no or least side effects, and it should not interfere with the sexual drive, desire, or the sexual act. Is that clear? Now let's talk about these contraceptive methods. Every year we are getting questions from this topic. See, first of all, you should know what are the natural methods. So natural methods are periodic abstinence, withdrawal method, and lactational amenorrhea. So the question was asked in 24-23. Now, periodic abstinence means abstaining from coitus from day 10 to 17. Why? Because ovulation is expected here. This is assertion and reason. Can we get such question? 10 to 17 day is considered as a fertile period. Now, second is withdrawal method, also known as coitus interruptus, in which partner withdraws his penis from the vagina before ejaculation. Third is lactational amenorrhea. Amenorrhea means absence of menstrual cycle. So, cycle does not occur during the period of intense lactation following parturition. So, see, this method has been reported to be effective only up to maximum period of 6 months. Well, that is just theoretical. It is not really effective for 6 months, hardly for 2 to 3 months. Well, that is the disadvantage of these methods. Advantage of natural method is there is no side effect because we are not using anything externally. We are just going in tune with the natural functioning of the body, right? So chances of failure, see, this method has side effects are almost nil. This is the advantage. The side effect or the disadvantage is, this is the advantage, and the disadvantage is the chances of failure are also very high. Is that clear? Now, talking about the barrier method, ovum and sperms prevented. What is barrier? Barrier means you're going to stop something from happening. You're putting a barrier in between. So, barrier between ovum and sperms from physically meeting. So, methods available for both males and females. So, how does barrier methods work? So, that mode of action is important for every method. So, barrier method also prevents fertilization. Well, same mode of action is in case of natural methods also. We are preventing fertilization here also. It works on the same prevention of fertilization. Is that clear? Works on the same way, in the same way. Now, condoms made up of thin rubber or latex sheath covers the penis in the male. This question was asked in Match the Following already. Now, it has to be used just before the coitus so that semen would not enter the female tract, right? So it prevents conception. Nirod is a popular brand name for the condoms for male. Now, the use has been increased to additional benefit of using the condom is protects the user from contracting STIs, sexually transmitted infections, another term for STDs. Is that clear? Both male and female condoms are disposable. These means one-time use only. Now they are self-inserted and therefore gives complete privacy. So this is the advantage other than preventing pregnancy, preventing the STDs. Another advantage, or why they are preferred so much, because they are self-inserted and can be used in complete privacy. Then there are other things like diaphragms, cervical caps and vaults. They are also barrier methods made up of rubber, only for females. Is that clear? And what do? They cover the cervix during the coitus by blocking the entry of sperm through cervix. But do these methods, diaphragms, cervical caps and vaults, prevent the entry of sperms in vagina? No. Sperm will enter the vagina though, but at the end of vagina where cervix begins, there diaphragm, cervical cap or vault is present if inserted, and it will not let the sperms to cross cervix. So they prevent the entry of sperms into the uterus, not in vagina. You want to prevent the entry of sperms in vagina, use condom or femidome. Are these points clear? Now, another effective popular method is use of intrauterine devices. They are placed by the doctor. They are not self-inserted. So, but the insertion is non-surgical. That is point. So, inserted by doctor or experts non-surgically into the uterus through vagina. And there are 3 types of IUDs. See, there are so many questions asked around IUDs. So, Lippes loop, copper releasing IUDs. So, Lippes loop is a non-medicated one, also known as inert IUDs. Copper releasing examples, CUT, copper T, copper 7 and multi-load 375. And the hormone releasing IUD, Progestasert, LNG20. Now see, they can give any number here after multi-load. So this number indicates the potency, how long this copper T can work, how many copper ions can they release. So number will vary, but multi-load itself means that it releases a big amount of copper ions. Are these clear? Now IUDs increase phagocytosis of sperms within the uterus, and copper ions released suppress the sperm motility and the fertilizing capacity of sperms. What are the 2 ways in which copper ions work? Suppress sperm motility and fertilizing capacity. So see, the question from this topic has been asked in 22, 19, 16, 14, 21, 17, 16, 23, 24. So there is hardly a year when NTA is not asking a question from this topic. Is that clear? Maybe in Re-NEET 2026 it comes back again. Let's see. So what about the hormone releasing IUDs? The hormone releasing IUDs make the uterus unsuitable for implantation and makes the cervix hostile towards sperms. 2022. Now IUDs are considered the ideal contraceptives and one of the most widely accepted methods. So which is the other most widely accepted method? It is condom. Is that clear? Now, oral administration, that we are going to talk about oral pills here, oral pills, oral contraceptive, right? So, either progesterone only or the combination of estrogen and progesterone. We never take only estrogen. Now, they are used in the form of tablets, hence pills. They are taken daily for a period of 21 days, starting within the first 5 days of menstrual cycle and after a gap of 7 days during which menstruation occurs, it has to be repeated in the same pattern. So roughly taken for around 21 to 23 days according to the female menstrual cycle, till the female desires to prevent conception. What is the mode of action? They inhibit ovulation and implantation, retard the entry of sperms. Now Saheli, the new oral contraceptive method developed by CDRI, right? It is once in a week pill with very few side effects, non-steroidal in nature and high contraceptive value. Is that clear? So which are the hormonal contraceptives? The question has already been asked. Which of the following are the hormonal contraceptives? So hormonal method is being used in lactational amenorrhea, intense lactation. It prevents menstruation or menstrual cycle. Why? Because during lactation, high prolactin, it is going to have negative feedback on FSH. It is going to keep FSH low. So low FSH means no menstrual cycle. Then pills, we have seen estrogen and estrogen progesterone combination or just progesterone. Implants, injections, and hormonal IUDs we have just studied, right? LNG20 and progesterone, all these involve the action of hormone. So this question was asked in 2019. See, which of the following methods are hormonal in nature? So understand the mode of action very clearly. This is the question. Is that clear? So look at the page. This whole page is so important, right? So progesterone alone or in combination with estrogen can also be used in female as injection or implants. By the way, injections and implants have a higher contraceptive value, right? And where are implants placed? Under the skin. So can we say subcutaneously? They are placed subcutaneously under the skin, and mode of action is similar to the pills, but effective periods are much longer, right? For 2 years, 3 years, 5 years, and so on. And as far as injections are concerned, you have to take injections once in a week or 15 days or once in 2 months, depending upon the potency of the injection, right? Now, very important, administration of these these pills, progesterone or progesterone estrogen or IUDs, within 72 hours of coitus has been found to be a very effective emergency contraceptive. Now, the difference between these oral contraceptives and these emergency contraceptives is these pills are taken regularly and before pregnancy happens or before coitus, right? These pills are taken post coitus when you're not taking using any contraceptive method. You're not on pills at all. But then if you fear pregnancy might happen, then this is taken post coitus, right? To avoid the possible pregnancy due to rape or casual unprotected intercourse. Then we have surgical methods or sterilization. This is known as a permanent method and only advised to partners as a terminal method, last method to prevent any more pregnancy. Now see, the question was vasectomy. No sperm in the seminal fluid but sperms present in the epididymis. See, vasectomy, in which vas deferens is cut and tied, the sperm production won't stop, just that sperm will not come out in the semen. So sperms are present in epididymis till vas deferens. They are present till here, but they will not come out because this vas deferens has been cut, right? So similarly, even after tubectomy, menstrual cycle will continue, oogenesis will continue, so menstruation will continue, right? So oocyte will be produced every month in a very normal way, just that the sperm, if enters, will not be able to meet or fuse with the oocyte. Is that clear? Hence no pregnancy. See, surgical methods, intervention blocks the gamete transport. So surgical method involves vasectomy, vas deferens cut and tied through small incisions on the scrotum. So it is not a major surgery, just a small cut is made through scrotum and it is cut. And then in tubectomy, a small part of fallopian tube is cut through a small incision in abdomen. So as compared to vasectomy, tubectomy is slightly a bigger surgery, but not major surgery. So generally it is the male who is recommended to get it done. Now, highly effective, but reversibility very poor, right? Is that clear to all? Now, okay, let's come on to MTP. So MTP is intentional or voluntary termination of pregnancy before the full term. It is also known as induced abortion. Learn the data. 45 to 50 million abortions performed in a year all over the world, right? And out of this 45 to 50 million, it accounts to 1/5 of the total number of conceived pregnancies in a year. So out of the total pregnancies that happen, 1/5 are aborted deliberately, right? Now, in India, Government of India has legalized MTP in 1971 with some strict conditions. Is that clear? And then there was this question. MTP Act, amendment of MTP Act in 2017, enacted with the intention to reducing the incidence of illegal abortion and consequent maternal mortality and morbidity. What does this Act, 2017 Act, MTP Act say? It says if pregnancy has to be terminated within the first, terminated within the first 12 weeks, then opinion of one registered medical practitioner is fine. You need to ask, just one doctor has to give his signatures and it is legal. But if pregnancy has lasted more than 12 weeks, that means after first trimester, but fewer than 24 weeks. Means if within the first trimester it is done, the recommendation of one doctor is legalized. Then if first trimester has crossed and it is between the first and the second trimester, means 3 months have gone but 6 months have not completed yet, so between 12 weeks and 24 weeks, more than 12 but fewer than 24 weeks, then we need the recommendation of 2 registered medical practitioners. Is that clear? Now, what are the grounds for any termination of pregnancy? The grounds for the termination of pregnancy. Number 1, it involves a risk to the life of a pregnant woman or injury of physical or mental health, and similarly, a substantial risk of the child born, physical or mental abnormalities. So, if it is harmful for the mother or the baby, in that case only it is allowed. Please tell me these points are clear. Right? Is that clear to all? So remember, MTPs are considered relatively safe during the first trimester, that is within 12 weeks only. Second trimester abortions are much more riskier. That is why we need a recommendation of 2 medical practitioners. Is that clear? Now, frequently if the fetus is to be, all right. Now moving on to STIs. Yes. So what are STIs? Sexually transmitted infections, also known as STDs or VDs, that is venereal diseases, or RTI. This is the same term. Then gonorrhea, examples: gonorrhea, syphilis, chlamydiasis, herpes, warts, trichomoniasis, hepatitis B, even AIDS. These are all STDs, which are completely curable ones. See, gonorrhea, syphilis, chlamydiasis, warts, trichomoniasis, these are completely curable. The question was asked in 2023. The examples were asked in 15. Which are completely curable? You must know, right? And which ones are completely not curable? Not completely curable. This was asked in 2024. Remember 3H. Just remember 3H. Hepatitis, herpes and HIV. These are completely not curable. Is that clear? So, how does it transmit? So, the mode of transmission is through sharing of injection needles, surgical instruments if not sterilized properly, with infected persons, transfusion of blood, and from infected mother to the fetus. Is that clear? So these are the mode of infections. So such questions are also being asked, right? Then causative agent of gonorrhea is Neisseria, syphilis is Treponema. Well, this was asked in 2017 though, but the syllabus has changed and trend has changed a lot since 2017. So you can learn it, but you can skip it as well. Now, what are the complications? See here. What are the complications here? The complications here are PID, pelvic inflammatory diseases, abortion, stillbirth means dead baby will be born, ectopic pregnancy, that means in fallopian tube or somewhere else other than uterus, infertility, and even cancer of the reproductive tract. So STIs are the major threat to the healthy society. Now learn this point, that it is most prevalent, reported to be very high in the age group of 15 to 24 years. So how can one be free from these infections? The same way you catch these infections, just avoid those things. Now what is infertility? So see, unable to produce children in spite of unprotected sexual cohabitation for even 2 years, cohabitation for at least 2 years. Still, if it doesn't lead to pregnancy, then this is called infertility. Now there is a technique through which a couple can have a baby. The special techniques are collectively known as assisted reproductive technologies, ART. So what are the techniques in ART? IVF, GIFT, IUI, AI, ZIFT, IUT. All the questions asked in 24. So which methods come under in vitro? So in vitro is these 3 methods, all in yellow. In vitro. Sorry, not in yellow. This is ZIFT and this. Sorry, all in green. These are in vitro. So in vitro methods involve embryo test tube baby, and embryo transfer. Is that clear? So whenever there is, and even ICSI. So three methods come under this. Actually, test tube baby and ICI, these are the two methods. Both these in vitro fertilization methods are followed by embryo transfer. Now the embryo transfer methods are of two types. One is ZIFT and another is IUT. It depends which stage you put back in the female body. Is it clear? If the embryo with more than 8 blastomere stage is transferred into the female body, it has to be kept in uterus. This is known as IUT. If it is less than or up to 8 blastomere stage, then it is kept in the fallopian tube, and this is known as ZIFT. Is that clear? So embryos transferred to assist the females who cannot conceive. This was a question. Which are the two techniques to transfer the embryo? It is ZIFT and IUT. It was asked here. Is that clear? Now, talking about in vivo. In vivo means in natural conditions. So fertilization will occur in the female body. Fertilization will occur within the female body. So these three methods are in vivo. Is that clear? So what is GIFT? Gamete intrafallopian transfer, where female can provide a suitable environment but cannot produce the ova. Is that clear? So intracytoplasmic, already told you. This is a method of in vitro where the sperm is directly injected into the ovum, right? Now, infertility cases either due to inability of male partner to inseminate or the female, or due to low sperm count ejaculate. So either there is inability to inseminate because of erectile dysfunction ED, or low sperm count. In both the cases, we have technique of artificial insemination. So guys, in this topic, you should know that under which situation we are going to perform a particular method. Is that clear? So when there is low sperm count or there is inability of the male to inseminate the female, in that situation we go for AI, or better method to AI is IUI. The difference is, in AI we place the semen in the vagina, and in IUI it is placed into the uterus. So better chances. Is that clear? Now, through the options, though the options are many, all these techniques require extremely high precision handling. So the question was asked in 25 about potential drawbacks of IVF. Number 1, high fatality risk for mother and the baby or the embryo if produced. First of all, for the mother, high fatality for the mother. So that is why it has to be dealt by extreme precision. Then by specialized professionals. Second, it is expensive, so not everybody can. And third, less adoption or orphans. And fourth, possibility of early embryo does not survive. So these are the 4 potential drawbacks of IVF. So these are the questions asked in 2025. Some general questions. Is that clear? So please tell me that these points are clear. And guys, with this, the first unit is also done. Yes, please. Let's move on to the next chapter now, that is of genetics. Is that clear? Quickly move on to the next page or the next chapter. Yes. All right, guys. Let's move on to the next unit now. The next unit is principles of inheritance and variation. Yes, all the serious students, give me a thumbs up. All ready for the next chapter? Yes, guys, all ready for the next chapter? You're feeling stressed. How can I increase marks? I have one way out. How can you increase your marks? You can increase your marks by taking these tests. You can increase your marks by taking tests every day. You still have chance, guys. You still have chance. Take these tests at your own convenient time. You want to sit at night and when everybody is sleeping and take the test, take it at that time. Subject wise, you want to practice more biology or more chemistry or more physics, there are 6 subject wise tests. You want to take overall syllabus test, you take this major test, and then full syllabus tests. There are 25 tests. Take this test series. Take mock tests every thrice. Now you have to take tests every day. More than this now, it is the time for these tests. Today is 12th. How many days are left? Take these tests every day. Analyze the test. Now, just reading the NCERT or attending these marathons, reading things blindly, and they won't help you much. Honestly, it won't help you much. Now read the things, work on that topic where you are weak. How will you find out which topics are you forgetting and which are your weak points? By the tests. And analysis is even more important. Analysis will take you around 1 or 2 hours if you do it properly. This is how you improve your marks. It's not about, me trying to promote this All India test series, but tests are important. What is a substitute for that? You are working with us. We are working together. Let's work towards this. You take test every day, every night. Take it twice a day now. One in the early morning and keep this time 2 to 5 certainly there. You take this test at least twice. Work on these minor tests, your subjects and major test and overall test. Analyze them. Make a list. Share it with us. Let me know in the comment section what are your weak points, where do you need help, what are your doubts. You can contact me on the Telegram channel and all the teachers. Talk to us. Let us know. If still it is not improving, then we'll talk again. But are you doing what you should be doing? No. There is no shortcut, guys. I don't have a magic wand. I'm not that fairy. I am not. You will have to work for yourself. If all the educators are working day and night for you, why are you not ready to work for yourself? You tell me if there is any other way out. You all have heard lots of things from the toppers, right? Every year toppers are giving some speeches and then they are starting their own things and telling you what they did, what they didn't. Have you ever heard any topper telling you not to work seriously and giving you any shortcuts? Have you seen any topper giving you shortcuts? Cuts are needed to increase your speed. And they are not really shortcuts. They are tricks. There is no shortcut. This is the way to improve your score. What else is the score improving method? You tell me. I will not say anything now. You tell me if there is any other way out. Tell everybody. And let's talk about now genetics. Alright, guys. Mendel, the father of genetics. How many characters did he study? A simple question. 7 characters. Which were the characters that he studied? The 7 characters studied by him: seed shape, seed color, flower color, pod shape, pod color, flower position, and plant height. The 7 pairs of contrasting traits in pea plants studied by Mendel. 2022 question. So, how many years did he give to the study? 7 years in 2017. What the years also should be clear. 1856 to 1863. And he proposed these laws of inheritance. Then, how many true breeding pea plant varieties were taken? These were statement-based questions. 14 true breeding pea plant varieties were taken as pairs which were similar except one character with the contrasting trait. Is that clear? Which experiments were conducted? Cross pollinations. Sorry, yeah. So Mendel conducted artificial and cross pollination experiments using several true breeding pea lines. How are true breeding pea lines generated? By continuous self-pollination to show the stable trait inheritance. Is that clear? So this is a process of artificial hybridization that he followed. Emasculation. We've done that artificial hybridization just now. Is that clear? Then coming on to what are genes? So genes, Mendel didn't use the term gene. He used the term factor. He used the term factor. Now we call them genes. Anyways, they are the units of inheritance. Contain information to express a particular trait. So genes code for a pair of contrasting traits. They are known as alleles. This was the lines, direct line in 2024. Is that clear? Now we know about this recessive and dominant. I'm not wasting time here. So a gene controlling one character, monohybrid. And when we are studying one gene inheritance, one character at a time, it is monohybrid cross. Is that clear? Then see, the production of gametes by parents, the formation of zygotes, F1 and F2 plants can be understood from a diagram called Punnett square. Who developed it? A British geneticist, Reginald C. Punnett. So where we can see the probability of all the possible genotypes and phenotypes as well. So this is basic. What is the genotypic ratio in monohybrid cross of F2 generation? This is monotype hybrid cross of F2 generation. Genotypic ratio is 1:2:1, right? What is the phenotypic ratio? That is 3:1. This is a phenotypic ratio, right? So basic. What is a test cross? See, Mendel crossed all the tall plants from F2 generation with the dwarf plant. This is called test cross. Test cross, in which showing a dominant phenotype is crossed with the recessive parent. What does it help? It helps us to analyze the genotype of the test organism. Who's the test organism? The dominant parent. What are the results of a typical test cross? See, a typical test cross, if the ratio is 1:1, that means half of dominant trait, half of recessive trait, ratio is 1:1, it means the parent was heterozygous, right? The organism was heterozygous. The dominant phenotype with unknown genotype, that was heterozygous if the ratio is 1:1 or 50%. If the ratio is, if all the individuals are 100% showing the dominant trait, it means the organism was homozygous dominant. Is that clear? So the numerical was asked in 2024, between black dominant over white, find the genotype of the black. And they had given you the value of the test cross. Now remember, back cross is a cross of F1 with any parent. So test cross is a type of back cross. So every test cross, see, every test cross is a back cross. But every back cross is not a test cross. So vice versa, not true. Are we clear? So ploidy of the chromosome sets in plants that you should know. Now, see the law of dominance. Characters are controlled by discrete units called factors. Factors occur in pairs, and one member of the pair dominates the other one. The one which dominates is called the dominant, other is known as the recessive. So law of dominance is used to explain the expression of only one of the parental characters in the monohybrid cross. So we know in F1 generation, it is the dominant trait that comes out. 100% progeny is showing the dominant trait, right? Now, law of segregation, which is, by the way, this is known as a universal law. And when does it apply? It applies during the gamete formation, right? So a homozygous parent will produce all gametes that are similar, while a heterozygous will produce two kinds of gametes in equal proportion. Now coming on to, there was a numerical in 2024 for a cross between pink and red, phenotype in progeny. What was the phenotype in progeny? If pink is crossed with red, it is showing incomplete dominance. What are the examples of incomplete dominance? Flower color in dog flower, also known as snapdragon or antirrhinium. So a cross between true breeding red flower and true breeding white flower, F1 gives pink, and when F1 was self-pollinated, in F2 generation there was this ratio 1:2:1. So this ratio, phenotypic ratio and genotypic ratio, same in case of incomplete dominance. While in case of Mendel, the phenotypic ratio was 3:1. Here it is 1:2:1. Well, this remains the same though. So R was not completely dominant over small r, which made pink to happen. Is that clear? Now explanation, see, what is the explanation here? The modified allele could be responsible for the production of intermediate feature. The normal, less efficient enzyme, non-functional enzyme, or there can be no enzyme at all. Is that clear? Now coming on to codominance. What is the example of codominance? ABO blood group, particularly AB blood group in ABO blood grouping. 2018 question. So what are the three phenomena which is exhibited in ABO inheritance? Number one, your multiple allelism. There are three alleles, IA, IB and small I. Then usual dominance as explained by Mendel and exhibited in blood group AB. Are we clear? Now, there is a numerical based question here also. How many genotypes and phenotypes are possible? This was asked in 2017. If this genotype crosses with this genotype. Is this point clear? Is this point clear to all? Is that clear? And now see this line, that in total 6 different genotypes are possible, 3 different alleles, 6 different genotypes, and how many different phenotypes are possible? How many different phenotypes are possible? 4. Phenotype means the blood group. 3 alleles, 4 phenotypes and 6 genotypes. These are the different combinations that are possible. Is that clear? Now can you draw a cross between this? Let me know the answer here. What will happen? How many genotypes and phenotypes are possible if this crosses with this? Can you quickly draw a Punnett square and see? Yes guys, give me an answer in the chat or in the comment section. What is the genotype? How many genotypes and phenotypes are possible if IA-IB crosses with IA small I? Well, the blood group here is AB and here it is A, but heterozygous A. This crosses. Yes, please. Let me know the answer. Now, in case of this B gene, this question was asked in 2019. There is one gene, starch synthesis in pea seed is controlled by one gene, which is B, and it has 2 alleles, capital B and small b. Now, starch is synthesized effectively by capital B, homozygous condition. So, it leads to large starch grains. If it is small b, it leads to smaller starch grains, and if it is capital B, small b, it is intermediate size of the starch grains. Is that clear? Now, therefore, dominance is not an autonomous feature of a gene or the product. It depends as much as on the gene product and the production of particular phenotype from this product as it does on a particular phenotype that we choose to examine. Is that clear? Is that clear to all? Is that clear? Now, inheritance of two genes. Now, inheritance of two genes, we are going to talk about dihybrid cross. So there was this numerical in 2022. What is the expected percentage of F2 progeny with yellow and inflated pod in dihybrid cross experiment involving pea plants with green inflated pod and yellow constricted pod? You have to solve this numerical. Let me know the answer, and if you get the answer as 18.75%, then you're doing it right. Yes, so this is the dihybrid cross, guys. What is another law which was depicted only during the dihybrid cross? That was law of independent assortment. So according to law of independent assortment, what ratio do we get? We get 4 phenotypes in the ratio 9:3:3:1. This was asked in 2025. Is that clear? Sorry. Take a screenshot here. Take a screenshot here, this numerical, and let me know the answer in the chat or in the comments later. Solve this question. If you are not able to solve, still let me know in the comment section. If you are, then you're doing right. Alright guys, coming to chromosomal theory of inheritance. Now see, I've already asked this question in one of my previous lectures, that who are the 3 scientists who rediscovered Mendel's work? So Hugo de Vries, the one who gave the concept of mutation, Correns and Sekmark, this rediscovered Mendel's result. Is that clear? Important names. Then in 1902, the chromosome movement during meiosis had been worked out. Walter Sutton and Theodore Boveri, they noted the behavior of chromosome was parallel to the behavior of genes, and this led to the discovery of or the development of another concept, that is chromosomal theory of inheritance. So see, here we say that Sutton and Boveri gave us the chromosomal theory of inheritance, and this was asked in 2022. But there is this important line: who united the knowledge of chromosomal segregation with Mendelian principles? That was Sutton and not Boveri. Please see that. Theory was put forward by both of them, but who united the knowledge? The answer has to be Sutton. So why am I bringing your focus here? Because there was this question in one of the books we were solving in the special class. So usually, we feel that, okay, these two have given this chromosomal theory, then both of them have united the knowledge, but no. When they are asking you who united the knowledge, answer has to be Sutton and not Boveri. Is that clear? Then this chromosomal theory was experimentally verified by Thomas Hunt Morgan. Thomas Hunt Morgan. So experimental verification was done by Thomas Hunt Morgan. This question was asked in 2020. And which was the experimental organism used? That was fruit fly, Drosophila melanogaster. Why did Morgan chose Drosophila melanogaster? For first reason, it is easy to grow on simple synthetic medium. Second, the life cycle is very short, which is about 2 weeks of life cycle. So you quickly get a new generation. Third, a single mating will produce large number of progenies. And fourth, there is clear distinction between male and female. That means sexual dimorphism. And so easily distinguishable. Is that clear? They are easily distinguishable. And fifth, many hereditary variations can be easily studied under very low power microscope. See, using electron microscope, involving that in study is actually full of a lot of hassle. So using electron microscope is a big deal, because there is a lot of process. Get the sample dehydrated, then run through the process, and then study it, get it gold plated. There are many things involved. But using a low power microscope is actually hassle free. Is that clear? So he used this fruit fly for his experiment. Now, during his experiment, Morgan also discovered another concept, and that was of linkage. See, he observed that two genes did not segregate independently. So Mendel's law of independent assortment, not for genes that are closely located on the same chromosome. So what did he say? That the genes which are very close by, they don't assort independently. Is that clear? So when the two genes in a dihybrid cross were situated on the same chromosome, then he saw a different result. Not the same ratio 9:3:1. He did not get that. Now Morgan attributed this due to the physical association or linkage of two genes and coined the term linkage. He gave the concept as well as coined the term linkage. Is that clear? Now, after Morgan, his student Alfred Sturtevant, he gave us a concept of genetic map. He said that the frequency of recombinants, the recombination frequency between the gene pairs, is a measure of distance between the genes. So see, normally when we get this percentage, we get this ratio 9:3:1. This 2 is the parental frequency. And these are the recombinants, right? Around 37.5 and this was 62.5. This is when independent assortment applies. But in case of Morgan, that is in case of linkage, what happened? He got another result. He saw that the recombinants, instead of 37.5%, he got recombinants only 1.3%. The frequency in which recombinants were obtained was just 1.3%. That's what led him to believe in the concept of linkage. See, so genetic mass, the unit used is centimorgan, and 1 centimorgan is equivalent to 1% of crossover, that is the recombination. So using recombination frequency, you can sequence number of genes. There is numerical based. So see here, in cross A and in cross B. In cross B, he is getting parental frequency 62.8, which is around 62.5, and recombinants as 37.2, very near to 37.5. So this cross is following independent. This topic, please go and watch the marathon. I have explained this cross in detail. What does each indicate? Is that clear? What does this yellow and white mean and wild type means? So please watch that in case of any doubt. Next, coming on to polygenic inheritance, guys. So again, it is a non-Mendelian pattern in which one character is controlled by multiple genes. It is also known as multiple gene inheritance. And see, human skin color is an example of this. Human skin color is an example of this. It is controlled by 3 or more genes. Such traits are polygenic traits. Polygene means multiple genes. Is it clear? Now, in case of human skin color, there are 3 genes involved, A, B, C. All are involved in the production towards melanin. So, effect of each gene is adding to the effect of the other. So, it is also known as additive inheritance. Is it clear? Now, so each gene will have its own allele. So, now in case all the alleles are dominant, that is this situation, it will have maximum melanin production and have darkest color, negroid color. And when all the genes are in recessive state, this will have lightest color, that means no melanin formation, albinos, right? And now when you cross them, you will see a different type of ratio coming out. So here, if any of the 3 recessive genes are, alleles are dominant or 3 are recessive, it will give you an intermediate skin color. It will give you an intermediate skin color. Is it clear? So here, it is not the quality, it is the quantity. So it is also known as quantitative inheritance. It is also known as quantitative inheritance. Is that clear? You can get a question out of this. When you're given 2 different genotypes, you have to cross and find out the phenotypes possible. So the trick is just count the number of dominant alleles. All the 6 dominant, darkest color. 5 dominant, very dark. 4 dominant, dark. 3 intermediate. Then after 3, if there are only 2 dominant or 1 dominant or 0 dominant, you get accordingly. And in my marathon, I have also given you the tricks, the formulae, how to calculate the number of those phenotypes. Is that clear? So learn those tricks for quick question solving. Now, what is pleiotropy? Where one single gene can exhibit multiple phenotypic expressions. This is 2023 question. What is an example of pleiotropy? One example of pleiotropy is phenylketonuria. Second example is also of sickle cell anemia. So sickle cell anemia, pleiotropy, these are both examples of and phenylketonuria, sorry, both are the examples of pleiotropy. What happens in phenylketonuria? Basically, there is an enzyme called phenylalanine hydroxylase. So enzyme is a protein formed by a gene, right? Now, if there is a mutation in this gene, which is forming the enzyme, what will happen? The amino acid phenylalanine will not be converted into tyrosine. So we need phenylalanine hydroxylase enzyme here for the conversion of phenylalanine to tyrosine. If there is a mutation in this enzyme, this will not be formed. What will happen? Phenylalanine will accumulate, right? So what will it lead to? It will manifest itself in mental retardation, reduction of hair, skin pigmentation, and even kidney damage. These are the manifestations of phenylketonuria. This mutation is called phenylketonuria. So basically what happens in phenylketonuria, there are multiple effects from one gene mutation. So one gene controlling multiple effects. So what is the first effect? First effect is that enzyme is not formed, and all these are the multiple effects, secondary effects caused by this mutation. Next is sex determination, guys. So different organisms have different types of sex determination methods. For example, insects like grasshopper, so they produce, their males show XO type. So 50% of the sperms have this structure called X, and other 50% did not receive this. Now this X structure was discovered by, this structure X body was discovered by Henkin. Is it clear? Later on it was found that this X structure is nothing but a chromosome. Now females in grasshopper, they have 2 X chromosomes. So accordingly, we know that it is always the heterogametic partner which is responsible for sex determination. So here it will the male grasshopper which will determine what will be the gender of the baby, right? Now see, grasshopper is an example of XO type of sex determination. This is asked in 2022. Now similarly, Drosophila. Now Drosophila, human males, we have XY type. We are XY type of determination, XY type of sex determination. Why? Because again the male is heterogametic, male is XY, and the female is XX. So the sex determination is based on the heterogamety type. So male XY, here male is XO. The questions have already been asked. Now in case of fowls or birds, here the female is heterogametic, so she produces 2 types of eggs, that is ZW. So female is ZW. Half of the eggs will have Z and half of the eggs will have Y. While here the female is heterogametic, hence female is going to determine the sex of the baby. So question was asked in 2019. Is that clear? For humans it is already clear. Now let's talk about sex determination in honeybee. In honeybee, the method employed is called haplodiplontic sex determination method. So what does it indicate? So female is diploid and 2n is equal to 32, while male is n and n is equal to 16. So female, that is the queen bee, the queen honeybee, she is going to produce gametes by meiosis. She is 2n. So gametes will be produced by, gametes have to be n, she will produce it by meiosis. Male is already haploid. So male will be producing sperms by mitosis. First difference here. First difference, male is haploid, female is diploid. Second, female produces gametes by meiosis, male produces it by mitosis. Is that clear? Now, what will happen? These eggs produced by the female, they can have 2 types of fate. Either this egg will fuse with the sperm, right? If it fuses with the sperm, it will lead to 32 condition. That means a female will be born. If the egg does not undergo fusion and directly changes into the new organism, that means without fertilization, parthenogenesis, it will lead to a male formation. So male is developed by parthenogenesis, no fertilization. Male is produced by parthenogenesis, while queen bee or the female is produced as a result of typical sexual reproduction. So the female can be queen or workers. So whether it will be queen or worker, that depends upon what it is fed at and what is the requirement of the hive. Are these points clear? Now let's talk about mutation here. So mutation is a phenomena in alteration of DNA sequences and resulting in change in the genotype as well as the phenotype. It leads to the variation in DNA. Is that clear? Now, this mutation can be due to loss or the gain of a DNA or some alteration in the chromosome. Now, chromosomal aberrations are observed commonly in the cancer cells. Now see, single base pair of DNA, if mutation occurs in single base pair of DNA, this is known as point mutation. For example, sickle cell anemia. Is it clear? Now, there are another kind of mutations, frame shift mutation. So what happens during frame shift mutation? There is deletion or insertion of base pairs of DNA. So now remember, if there is, wait. See here. So if there is one or two base pairs, if one or two base pairs are added or subtracted, remember, one or two base pairs are added or subtracted, it leads to frame shift mutation. But if three base pairs are added or subtracted, plus minus means three base pairs are added or subtracted, then it will not lead to frame shift mutation, no frame shift. Although there will be addition of one amino acid in it, but the frame will not shift. See here. And one more thing, the chemical factors or the physical factors which induce mutations, they are known as mutagen. It could be some radiations also. Is that clear? Now, genetic disorders, pedigree analysis. What is the pedigree analysis for? So it is, a pedigree analysis is a tool. It is a tool to analyze for the analysis of a trait in several generations of a family. So it is a genetic tool to study the inheritance of a particular trait. Is that clear? To see the possibility of inheritance, whether it will be transferred to the next generation or not. So it is studied by a family tree. So it is a strong genetic tool. Is that clear? To study the inheritance, especially for the diseases. Now, let's talk about the Mendelian disorders. Mendelian disorders are of two types, guys. One is the genetic disorders are of two types. Wait. Yes, genetic disorders are of two types. One is the Mendelian disorders and second is the chromosomal disorders. So what are Mendelian disorders? It is alteration or mutation in a single gene. Is that clear? It is increase or decrease, yani alteration or mutation in the single gene. And now this can be traced by the pedigree analysis. For example, we have hemophilia, cystic fibrosis, sickle cell anemia, color blindness, phenylketonuria, thalassemia. These are all Mendelian disorders. Is that clear? So they can be dominant or recessive. Clear to all? Now out of these, hemophilia and color blindness, these are X-linked. See, hemophilia is X-linked recessive trait, right? And pedigree analysis will be discussed in a while. Color blindness, this is also sex-linked recessive disorder. And which one? X-linked recessive. These are the questions that have already been asked. So what happens in color blindness? Red-green color differentiation is not possible in the person. So this gene is present in the X chromosome, so occurs about 8% in males and only 0.4% in females. This is because genes lead to males have only one X chromosome and female have two. So this is the assertion and this is the reason why it occurs more frequently in males, because male have only one X chromosome. So it will be expressed there. In females, this trait, color blindness or hemophilia, will be expressed only if She's homozygous for it. Is that clear? Now, there was a numerical asked, colorblind female marries a male whose mother was colorblind. So what are the chances of the progeny being colorblind? You have to find out. So I have discussed these questions in my special classes. I am still discussing. Please make sure that you join me. Then, woman with X-linked condition on one of the X chromosome. This chromosome can be initiated by both sons or daughters, or by one son and daughter. So this was question asked in 2018. So please make sure that you're solving these questions with me. Hemophilia, again, sex-linked recessive disorder. So transmission from unaffected carrier female to some of the male progeny. A single protein that is formed, cascade of proteins and involved in clotting of blood is affected. So a heterozygous female is known as a carrier. She can transmit the disease, but herself she is not hemophilic. So possibility of female becoming hemophilic is extremely rare because mother of such female has to be at least a carrier and father should be hemophilic. Very important line, assertion reason, right? Also known as royal disease because it is found in the family of Queen Victoria. Sickle cell anemia, what type of disease it is? Autosomal linked recessive disorder, first of all. Now, controlled by single pair of allele. So there are these two conditions. So in this case, heterozygous case, partially the hemoglobin will be normal, partially it will be affected. Now, individuals with this show the diseased phenotype. Heterozygous appear apparently unaffected, but they are carriers. So 50% chance probability, right? And those which HbA are absolutely normal. What happens in the sickle cell anemia? So GAG is replaced by GUG. So there is a single pair. It is a point mutation, right? Just one base pair change. A to U, GAG becomes GUG. So instead of glutamic acid, it is replaced by valine. The whole beta chain of hemoglobin is destroyed. So this is normal hemoglobin, this is sickle shape, and see, it affects the structure of RBC also. Is that clear? So solve these questions, guys. Colorblind male marries a female homozygous for normal vision. So homozygous for normal vision is XX, and colorblind male means XCY. So can you make a cross out of this and find what is the probability of the son being colorblind? This was a question in 2016, right? Then phenylketonuria. It is again an autosomal recessive disorder. I've already told you what happens in this, right? Then thalassemia was asked in 2020, again autosomal linked recessive disorder. Now these are of three types basically, but here it is mentioned too. Alpha thalassemia, in which alpha globin chains are not produced. In beta thalassemia, beta globin chains are not produced. See, alpha thalassemia is linked to genes HbA1 and HbA2 found on chromosome 16. This has already been asked in 2024. In beta thalassemia, the chromosome number 11 is affected and the gene involved is HBB. So this line, thalassemia differs from sickle cell anemia in being a former, that is thalassemia is a quantitative problem of synthesizing the few globin molecules, while this is sickle cell anemia is a quantitative problem with incorrectly functioning globin. So all these lines are PYQs. Please let me know if there is any doubt. Then guys, we have chromosomal disorders. Now chromosomal disorders are due to excess or abnormal arrangement of one or more chromosomes. Either there is one more excess or there is one less chromosome that leads to chromosomal disorders. So this condition is known as aneuploidy. For example, Down syndrome due to extra copy of 21st chromosome. Now why does this one excess come along or one less chromosome is present? This is because of non-disjunction during anaphase. Do you remember? Separation of sister chromatids, right? So that separation doesn't occur. So both the chromosomes tag along. So non-disjunction. So due to the loss of X chromosome in this is Down syndrome, one extra copy. Turner syndrome is due to the loss of one X chromosome. So while the normal females carry two X chromosomes, Turner females carry only single X chromosome. So this is also due to failure of cytokinesis after telophase stage of cell division, results in the increase in whole set of chromosomes in, so this will cause polyploidy. See, we have two types of chromosomal disorders. One is aneuploidy, second is polyploidy. In aneuploidy, only one or two chromosomes show one less or one more copy, extra copy. But in polyploidy, the whole set increases. Why will the whole set increase? When cytokinesis does not occur after the telophase. Are you getting that? So when cytokinesis will fail, there will be whole increase. So there will be increase in the whole set. This is polyploidy. We don't get to see polyploidy in humans or animals because such embryo will not exist. But it is seen in case of plants though. Right? Now, these are the important terms. So important features. This is in Down syndrome. What happens? Broad flat face, big and wrinkled tongue, congenital health disease, flat back, loops on the fingertips and palm crease, right? And then we have Down syndrome. So this is studied by Langdon Down. All these things have been asked. See, all the factors, all the symptoms, who studied it, what happens. These are important. 23-24 was all about Down syndrome and Klinefelter's as well. In Klinefelter's, there is additional copy of X chromosome. So while the normal male is XY, Klinefelter's male is XXY. So total chromosomes 47, overall masculine with feminine development, like development of breast, called gynecomastia. So they are sterile. So there were questions asked. Question has not been asked around Turner syndrome. So let's expect this question in 2026 Re-NEET. So disorder, absence of one of the X chromosome. So female is short of one X chromosome. Total chromosome number is 45. So again, sterile, ovaries rudimentary, features, she lacks some secondary sexual characters. So overall masculine features are present in Turner syndrome. Is that clear? So guys, with this we complete genetics. We have completed 4 chapters. I know at 7 o'clock you have another class. So I'll be stopping now. We'll take another part 2 where we'll be completing rest of the chapters. Is that clear? So you'll be informed. Make sure that you are studying these questions, and most importantly, you are taking these tests. Without taking these tests, revising NCERT is a futile effort, to be honest. So if you are not taking any test, you're not practicing the questions, and just practicing at your own luxury is not helping. Under strict conditions, time bound. So you have to practice these questions. And again, I'm telling you, don't prepare yourself according to the luxury of extra 15 minutes. No, they are to complete the formalities. And it is just NTA trying to gain your trust, trying to keep you happy. You keep yourself prepared. You be prepared for 3 hours, rather 2 hours, 45 minutes. Right? If you are able to manage the time and show efficiency in this time period, then you are on the right track. And how do you practice that? You have to revise these things. You have to practice these things till the last moment at your own convenience. Home tests, and analyze them. You need solutions for that. And the tests should not be random. You don't have to practice any random question. You have to practice NEET oriented questions. Right? Take this test series. Let me know your results. Let me know what are the challenges you are facing from these test series.